Good afternoon, everyone. Welcome to the Brainstorm Summit. Yeah. This is our second year, and we are growing by leaps and bounds. I want to make sure you know that we have lots of great sponsors in this room as well. So during your breaks, make sure you get to see what's going on there. They're made sure everything's really fun and relevant. We also are going to have some sessions across the hall at H&J and at K. We have a live stream going out here in this room, and we have one going out at H&J. We have kids that are upstairs. If you have kids and you haven't found us, we are in Hart, Russell, and Cannon. And those activities this afternoon will run until 5:30. Bring your kids down to enjoy the reception. And then tonight is Dad's Night. And that's going to be at a fun virtual golf place called 5 Iron. There'll be some additional food, drinks, dads, and men in general. Go have some fun. And women, stay with me because we're going to have a women's fair. It's going to be fun, casual, more time to talk, drinks, and lots of fun activities. So I want you to be a part of it. If at any time you need something, stop at the registration desk, grab Jenny, who is my sidekick, Jenny McLean. She works with Kat.
It is packed in here.
Wow,
this
what
is
is
amazing to
this?
see all the people from all over the country coming together.
James
This
Origa, is
awesome.
that you?
Yeah, it is. Hi, Lisa.
Hi.
Again, sorry for the interruption. Sorry for the logo. I can and will fix that.
James.
We've been creating something really special, and I want to show it to you. So take a look.
Okay.
I've had the honor of working with some of the most incredible kids and teens who are impacted by pediatric cancer through our Big Dreams program with the Children's Cancer Research Fund. We focus on bringing patients' imaginations to life in unforgettable ways, whether we're making their own animated music video, planning community-wide live action events, or developing their own custom video games. Our focus is always on the child and how we can make their dreams not just dreams. The results have been outstanding, and their creativity has inspired thousands. Recently, I shared stories with a high school in New Hampshire about pediatric brain cancer, and in particular, DIPG. And the next thing you know, it led to a beautiful group brainstorming session. It focused all about how we can gamify DIPG in order to educate others in a way that has never done before. And thanks to augmented reality, now you can place a giant brain right in front of you. You can walk inside of it and physically grab broken neurons to collect educational cards. It's pretty incredible, and it's just the start of what's possible. Each one of you will be given a QR code so you can try.
Gentleman, James Origa.
All right. Sorry to interrupt. There was just so much action-packed with putting a giant brain in the room that it just had to happen. Hi, everyone. It's so good to be here with you all today. Hopefully, by watching that video, you can see a little bit of the magic that we've been able to work and create with patients from all over the country. Some of you here, I've worked with your kids and helped them create their own video games and all sorts of magic. But one of the things I wanted to highlight today was that idea of how we put a giant brain using your cell phone to create something really incredible, to make DIPG and bring this type of awareness as like a video game. You know, how often, how unique is that? Because no one is doing that yet. And how powerful was it? You could see all these students working together because they saw, when I talked to the school, they were so excited about how can we help? They're not scientists, they're just students. And so they were like, well, we love playing video games. Let's make it happen. So next thing you know, you have a giant brain that teaches you all about DIPG. You have to grab the neurons, connect it. And now this game has gone global, and it's all used with just your cell phone. So with this simple tool on Snapchat, what you can create, and now it's in the hands of all these people. It's gotten the attention from Snapchat, Meta, all these people from all over the country and all over the world now have DIPG on the map. In a really, really unique way using your phone. And some of the stuff, how many people here have even heard of augmented reality before? Okay, yeah, slim pickings, but we got a couple. That's good. So that's the point. We're trying to get creative with this tool, and it all started because of this little boy, this teenager named Gavin, and some of you all may recognize him and worked with him as well. But Gavin, he was going through his brain cancer and DIPG, and he was getting so frustrated because he couldn't play video games anymore. And he got so fed up thinking about other kids like him that are going through the same process that are missing out and can't play games. So we started thinking and brainstorming, what can we do? And he was, even though he was in pain and going through so much stuff, he wanted to focus on others. And so many of these kids often do so, but he wanted to create a game for other kids like him. So we started brainstorming, thinking of, you could use Instagram and these filters in really unique ways. So he ended up creating this awesome video game where he could drive his race car and collect a cure for DIPG. And it was just a way to get education and awareness out there and get people talking about this. And it got involved with the Fast and Furious and all sorts of things that happened for Gavin. And now his legacy is still preserved by seeing thousands of people from all over the world playing his game. Same with Esther, and when she wanted to collect kittens from the snow, and her mobility was limited, so we were able to use head tracking so she could lean left and right and steer their character. Same with the cars. So it's just, how can we get creative with these tools? I'm showing you what we're creating with just a cell phone, but we're in a room here surrounded by the brightest minds, scientists, the biggest-hearted folks here, each one of you, and I'm just sharing these stories with you to hopefully spark some excitement and spark some new ideas of what can you create. All of this beautiful magic started with a brainstorm, and we're here with a couple of days to brainstorm with each other, not just learn about science and all the magic that comes from that, but take your experiences that you've been through with each other and turn that pain into fueled passion and see what you can create. Because hopefully some of this stuff can kickstart your week here. And like I said, this is an actual video game that is played on Snapchat. So I want you all to try it. We're going to be passing out cards here by the end so that way you can all try it. But heads up, it's difficult. Just like these diseases and these cancers that we're trying to fight, it's difficult. It's really hard to solve. So you're not going to quit, though, if you can't figure it out the first time and get all frustrated. Yeah, it's going to be frustrating. But you're not going to give up. You're not going to quit, just like what we're all doing here today. We're working together as a team. We're brainstorming. We're coming at these problems in different ways than what people are used to. So be creative in your problem solving. Be creative with your time here with each other. Enjoy this time with each other. And thank you, Lisa, for letting me interrupt and jump in here. And thank you all for having me. And hopefully this sparks some ideas, because I want to show the world how powerful a brainstorm can really be. So thank you very much, and I'll fix that logo real quick for you. Sorry, I got to put that back. All right, there you go. All right.
Isn't he amazing? Thank you to Big Dreams Tour. Thank you for the Childhood Cancer Research and James Origa and Lad in the Battle. And if you are joining the Tough Together online community, you'll see that James is standing up a little fun spot there to connect with kids and do stuff. So as you work with families, you'll know that. I just want to thank our sponsors. I want to thank our speakers. I want to thank an amazing committee who has worked very hard. In particular, I want to thank V and Annette for their program and signage design. I want to thank Sylvia and Reagan, who have put together so many things, including the decor at our luncheon and just getting all the volunteers ready. I want to thank Samantha Chacon, who's behind the scenes making AV work, Elizabeth and David Turner, the same, and Sarva, who put together a number of our panels. And then we could not be here if it weren't for Misha Mehta and Jenny McLean, who have worked 18-hour days, a long, long time, alongside of me. I am forever indebted. As you saw, we want you to be casual. We want you to have fun. James kicked it off in just the right way. We hope that what you'll hear today are some really new innovations, some new research, some new inspiration, some new ways to connect with families. We want you to bring your ideas as well. It doesn't take a special recognition to have a seat at this table, because we need everyone involved. We must work faster. We must work together across all tumor types in this pediatric brain cancer community. So if you don't see, for instance, someone that you want to be here because they are the person for the tumor type that you're involved with, then we need you involved, because we might not know that person. And we're going to ask you to get involved next month and help us plan for next year. I want you to forget all about the status quo. I want you to forget all about the barriers, whether it's funding, politics, institutional barriers, that feeling inside of you that says, I shouldn't, I can't, nobody asked me. And I want you to give us the next 3 days to think, but what if I did? What if I could? What if I asked? What if I spoke up? What if I decided that I only had a year to make an impact? What would my impact look like? And would it be something within pediatric brain cancer? Because if it would, I'm just going to ask you with me to say, what if I would be as brave as the kids I'm fighting for? You guys, that's exactly what it's going to take. Because they don't have any way to say, yeah, not today. I don't think it's going to work for me. They just have to push so hard, don't they? But what if we did too? What if we did that together? And what if we didn't worry about what somebody else thought? But we just found ways to move the needle inch by inch by inch until someday, in the words of my friend Adam Resnick that I quote all the time, because it was so dear to me when he sent it to me in a text 2 years ago when we started the Tough Together breakfast. He said, what about that day when we will no longer be afraid of what cancer can do to our children? Instead, we will celebrate what we did when we came together and changed history. Our job is to do that now and to do that faster. And there is not one person too many in this room to get it done. We need you all. Ginny, what have I forgotten that they need to know?
Wow, how do you follow that heartfelt? So I'm here to follow a little bit more of James, and we're all going to have some fun here. So we have the best speakers, we have the best researchers, we have the fabulous sponsors. We have family advocates that have been in this for 10, 15, 20 years. We have family advocates that have been in this for 10, 15, 20 minutes. Make old friends your new friends. Get together and take pictures for us. We want to blanket social media with Brainstorm Summit, and we want to thank our speakers, get pictures with the speakers, tag them, Brainstorm Summit. Talk to our sponsors. They have some amazing things in store for this community. Take pictures with them, post it. We want to share the message of our sponsors for the next 3 months as we get you all involved in building next year's Brainstorm Summit. But take time to talk to each other, engage with each other, learn from the folks that have been around a long time, be inspired by the folks who are new, and always, always remember our kids, because that's why every single one of us are here. So I think I'm getting Misha up here.
Yeah.
So post, tag, and let's get social media flooded. And Misha, where's Misha? There she is going to come up and introduce our first keynote speaker of the weekend. Oh, I missed something? I forgot something.
I just wanted to lean in real quick. If there are people in the back of the room that need to come up to the front, it's like church. You accumulate, you get the front pew. That's just fine. And to our speakers, if you're in this room, you'll have a confidence monitor down here with a timer. So just kind of keep an eye on that, okay? Thank you so much. Watch your program so you know when we split apart and we have more options for you. Thanks, you guys. Have a great program.
Good afternoon, everyone. It is an honor to introduce our first keynote speaker today, Dr. Elaine Martis. Dr. Martis is a luminary in the field of genomics. She is the co-executive director of the Institute of Genomic Medicine at Nationwide Children's Hospital and the Nationwide Foundation Endowed Chair in Genomic Medicine. Dr. Martis is also a professor of pediatrics at the Ohio State University College of Medicine and has authored over 350 different peer-reviewed papers. She is the past president of ACCR and was also been elected to the National Academy of Medicine. Under her leadership, Institute of Genomic Medicine has partnered with NCI and Children's Group of Oncology to produce clinical results for pediatric patients for comprehensive profiling to populate the CCDI database to enable future discoveries that will improve pediatric cancer outcomes. Dr. Martis' contribution extends beyond the lab. She's a champion for scientific collaboration and an advocate for responsible genomic research. Ladies and gentlemen, please join me in welcoming Dr. Elaine Martis.
Oh, and I have a gift waiting for me here. Thanks so much, Misha, for the nice introduction and for the opportunity to address this group today about pediatric brain cancer, which is my topic on genomics of pediatric brain tumors. It's a real honor to be here, and I'm happy to get things started. So I want to, recognizing that there are lots of non-scientists maybe in the room, wanted to get things started at a very basic level and work our way up so that everyone sort of starts on the same page. So one question you might be asking yourself is, okay, pediatric brain tumor genomics, what does that last word mean? The other ones you probably are unfortunately well aware of. So what is a genome? If we look inside of ourselves, a very complex organism, we are made up of billions of cells. Each cell has a nucleus, and these little X-shaped guys inside of this sort of caricature of a nucleus are chromosomes. And of course, encoded on the chromosomes is essentially the book of life, how humans develop from a fertilized egg, a miracle, really, that it ever works out well, if you ask me, and I've had lots of exposure to when it doesn't work out well. So inside of the chromosomes, along their length, rather, are genes, and these genes encode the proteins that are in our cells and are the primary building blocks of the cells in our body, proteins. There are obviously other important components, but they're the major ones. And it's this interesting sort of encoding of proteins on the genes that happens with only 4 basic subunits, A, C, G, and T, the 4 nucleotides. Now, if you think about binary code, which is what makes your cell phone work and your laptop, et cetera, and stores data, we actually have a better code. We have a quaternary code. But we also know that when one of these bases gets disrupted or changed, whether it's in the course of development as a germline or constitutional mutation, or in the course of changing a normal cell to a cancer cell, there can be problems that are introduced because of that change in the DNA code. And what I'll talk about today is the way that we use the DNA code, which is simple and rapid to decode, to really interpret what changes go on at the level of proteins, which is really where many of our therapeutics and other approaches are focused. So let's move to the next aspect of the genome, and this is, again, one that we're all painfully aware of. Cancer is a disease of the genome. So this has actually been a fact that was well before we understood ACs, Gs, and Ts, or even the structure of chromosomes, predicted from early thinkers like the ancient Egyptians. Sorry, I'll get my buttons right here in just a minute. Ancient Egyptians who were sort of thinking about cancer and first describing it about 5,000 years ago. An early German zoologist in the 1900s first observed altered chromosomes in plants and thought about how changes in those structures, which he didn't really understand what they did, might actually lead to changes in the cells. And this was the first sort of recognition that chromosomal changes might be the basis of cancer. And then one of my scientific heroes, Dr. Janet Rowley, is pictured here. In the 70s, as a pathologist, a rare woman in medicine, even at that point in time, which for somebody my age doesn't seem like all that long ago, she essentially was investigating technology to get chromosomes to condense and stain so that she could see them better under the microscope. And once she worked out that technology piece, then they became obvious under the microscope, and she systematically worked through acute lymphocytic and myeloid leukemias by looking at chromosomes of patients that were similarly diagnosed and figuring out that actually there were changes in the chromosomes that could be identified. This is just a fuzzy picture from one of her early papers in the 1970s, first describing the canonical translocation that occurs between chromosome 15 and chromosome 17, putting two genes together called PML-RAR alpha. And this is a definitive hallmark of a specific type of acute myeloid leukemia called acute promyelocytic leukemia. Actually, one of the most curable leukemias that exist today. But that fundamental knowledge that she introduced sort of led the direction in why this chromosomal change actually occurs and what it means for the patients and how they're treated. Sorry, my badge is giving me problems here. So, this is the basis of cancer as a disease of the genome. And now we want to talk a little bit about genomics and what genomics is. You may have guessed already, or you may already know, but it's the study of genomes. And here we principally focus on unique DNA sequence alterations in the context of those proteins they encode and what those proteins function to do in the normal cell and how they're changed in their function in the cancer cell. And part of the secret sauce here is that DNA sequence gets copied to an intermediate called RNA, and then that RNA gets translated into proteins. And so we can easily interpret when we have changes in the DNA what might change in the sequence of the protein. Part of the challenge in genomics, of course, is that then figuring out what that does to the function of the protein is often not well understood just yet. And we are developing new high-throughput methods to do that, we and others. But that's really still one of the remaining challenges in this genomics equation that I'll talk about today. Ultimately, then, we have to figure out what the changes mean, right? And so one of the things I really want to emphasize today, and you're probably already aware of, is we have to interpret the changes by looking at databases of results from other large-scale studies and from case studies, other patients that have been sequenced and understood before in the context of that particular mutation in that particular gene and what role it might play in cancer. And so the bottom line is that sharing data from cancer genomics studies is really important. I'm not the only person you're going to hear that from this next couple of days, but I just really want to emphasize that everything we do, whether it's the large government program I'll talk about today or in our own cancer studies of our patients at Nationwide Children's Hospital, all the data that we generate is deposited into publicly accessible databases. All of the publications that we produce and publish in the peer-reviewed literature are produced as open source information, so you don't have to have a subscription to the journal in order to get the paper and download it. So this is a real commitment, and I think it's a commitment that makes sense, because at the end of the day, pediatric cancer is a rare disease, and the only way we get ahead is by sharing data and information. And lastly, genomics is driven by technology. I've essentially been involved in technology development around genomics since I was a lot younger than I am now, early graduate school. I learned about DNA sequencing from a professor, my major professor for my PhD, who learned DNA sequencing in the lab of Fred Sanger. And if you don't know who Fred Sanger is, that's okay. You can look him up on the internet. He won the Nobel Prize for figuring out how to sequence DNA. So I always like to say I'm 2 degrees of separation from Fred Sanger. Back in the day, I was part of the Human Genome Project, decoding the original template that we compare all genomes to nowadays. It was a very large international project. We used large sequencing instruments like the one shown here, and at the peak of the Human Genome Project, we had about 150 of these instruments in our laboratory at Washington University in St. Louis. We were cranking out data all the time and depositing it every night. And what resulted was the human genome, along with multiple other labs around the world. Obviously, this was a large international project. But if we had to go back today and use these same instruments to sequence individual human genomes, like I'll talk about today, it would be a very expensive proposition, sort of a moonshot, if you will. We can't do that anymore. But the good news is there's technology. So this may look like a washing machine to you, and it looks like a washing machine to me. I call it the $1 million washing machine because that's essentially how much each one of these instruments cost. However, they don't cost $15 million to sequence the human genome or resequence it. This is a little bit out of date. This is last year's technology. We're just getting the newest NovaSeq instruments in our laboratories. But the bottom line is that you're at about $600 to $1,000 per genome. And look at the rate of data production. So 24 whole human genomes just in a couple of days. The newest instruments will do it in 24 hours. So this is a pretty remarkable change and puts us in the realm of clinical sequencing, which I'll talk about today, as being a real thing that's affordable, that is a rich information source, and is going to drive the innovations of the future for cancer patients all over the world. And this we refer to as next generation sequencing because the technology is new and significantly different from this old box over here. So I'm going to talk a little bit now about setting the stage for pediatric cancers with setting the stage for adult cancers and sort of explain to you what the current reality is in terms of a highly prevalent cause of cancer that has been studied widely because lung cancer patients in the adult setting are fairly easy to find. There have been a lot of clinical trials as a result of that. This is the top most diagnosed cancer in the United States in men, and unfortunately the second most frequently diagnosed cancer in the United States in women. So we know a lot of information about this cancer, and it's really changed just over the last 15 years or so from pathology-based evaluation under the microscope, which says this is an adenocarcinoma or a squamous cell carcinoma, to the combination of pathology, which is always important, and genomics, which has told us about the predominant driver mutations that occur in non-small cell lung cancer, the most prevalent type adenocarcinoma that's diagnosed in individuals. Well, what has this meant? If you know anything about therapies and genes, you can kind of look around this circle and say, hey, I've heard of epidermal growth factor receptor before. I've heard of KRAS. These are all genes that drive lung cancer. And as a result of knowing those genes, pharmaceutical companies have been extraordinarily busy finding drugs that are highly specific and target these genes and the mutations that drive the cancers. And so the reality for patients nowadays diagnosed with lung cancer is that they will have a biopsy evaluation of the cancer, and from the biopsy material taken out of that lung, we will perform next generation sequencing based analysis and identify which of these mutations are involved in their cancer. And that will give us answers about how best to treat these patients. Even in the first line, even at their first diagnosis, you now see patients with EGFR mutations experiencing a number of different possible therapeutic outcomes. These are all targeted therapies, they're all highly effective, and they have low side effect profiles. No more chemo, no more radiation. EGFR and KRAS mutations are frequently identified in smokers who, because of cigarette smoke and its very prevalent capability to create mutations, have a very high mutation load. And we know from clinical trials that these individuals will respond well to immunotherapies, checkpoint blockade immunotherapies in particular, with or without chemotherapy. So this is the current standard of care in this setting. And then we get to gene fusions, which I already mentioned for acute promyelocytic leukemia. There are also ALK drivers in lung cancer, and these have a variety of different drugs that are already FDA approved as well. Highly effective, low side effect profiles. So question one is, how do we get to this with pediatric brain cancers? Well, here's another fact that all of us are painfully familiar with. Leading cause of death by disease in children, and more frequently diagnosed now than leukemia. So this is a continuing problem, and in many cases, as we already also know, the standard of care has not changed for many of these diseases in over 40 years. We see cancer incidence rates here, just to support what I just said about the prevalence of CNS malignancies being diagnosed in our children. And we also know from early cancer genomic studies in the pediatric setting, here's the Pediatric Cancer Genome Project, a 5-year, $60 million program that we carried out at Washington University with St. Jude, looking across different tumors from leukemias, well, I didn't do that, to solid tumors to brain tumors. We were able to identify some facts, and they're sort of listed here. few point mutations, gene fusions that drive cancer onset, characteristic deletions or amplifications on specific chromosomes that can be tied directly to disease outcomes. I'll illustrate all of these points. And then finally, we know that, in general, pediatric cancer shouldn't happen. Cancer is a disease of old age, accumulated mutations through many cell divisions and mistakes that happen, but of course they are a reality. And so we understand, but probably not completely, the entire possibility list of genes that are driving predisposition to cancer, and I would argue this is best studied by the normal genome. Usually a non-disease involves specimen like blood or buccal swab. So this is sort of setting the stage. The other component that's known and is, again, unfortunate in that setting of drug development for, high value proposition, if you're a pharmaceutical company, is that if we look at the overlap between genes that are significantly mutated in adults and significantly mutated in kids, there's a pretty small overlap in that Venn diagram. There aren't a lot of shared mutations, but there are a lot of unique mutations in the pediatric setting that still lack for suitable therapies to address those as drivers in the cancer. The last thing I want to mention is survivorship, which continues to be a challenge, and this is really a rationale for using more targeted therapies when that's possible. Why? Because we know that if you look over the ages from kids diagnosed and treated in the 70s, this is really sensitive, sorry, 70s, 80s, or 90s from the study in JAMA a couple of years ago, we haven't really moved the needle so much in terms of long-term sequelae. Now, being a cancer survivor is a good thing to be able to say, don't get me wrong, but we know that these survivors through the decades have excessive downstream sequelae that are negative consequences of aggressive chemotherapy and radiation therapy that lead to challenges in adult life that affect quality of life and also lead to high secondary risk of cancer occurrence. And so this is really a reality that I think what I'll talk about today is committed to changing. So the complexity of pediatric cancer brings us to this notion that diagnosis really needs to be what I call multi-omic. That means we need to look at a number of different assays, which I'll tell you about today. So pathology, of course, is a critical starting point, but we know in pediatric CNS malignancies in particular that about 10 to 15% come through pathology evaluation with an indeterminate diagnosis. That's a fancy medical way for pathologists to say, I'm not really sure what kind of a cancer this is. And we know that we need additional information to really help define that, and sometimes that information can come from the molecular profiling that I'm talking about. Also, there are some inroads to targeted therapies for pediatric cancers, which I'll just briefly describe. We want more, but we also know that we have to have that molecular profile to get to that point. So for brain cancers, these indeterminate diagnoses may be informed by an assay called methylation profiling. I'll describe that in just a minute. But we know that methylation profiling is so important that in 2021, the World Health Organization included it as a component of diagnosis for pediatric brain cancers. That means it's important, and it's important to carry out, and it's actually straightforward to carry this out. We also need to test for known fusion drivers. Why? Sometimes they're diagnostic, but increasingly we're seeing that some percentage of them are also therapeutically addressable if they're drivers for the cancer, and I'll show an example of that. And then finally, the most complex but as informative component comes from comparing the DNA from the cancer to that of the normal or non-disease affected tissue, such as blood or saliva. What do we learn from that? Many things. Specific chromosomal features that tell us to dial up or dial down the treatment or therapy that's being used. Specific gene mutations that might identify a targeted therapy. And more recently, you may be aware that in very rare setting that affects some kids with brain cancer diagnosis, such as constitutional mismatch repair defect, Those same immunotherapies that are effective on lung cancer patients that are former or current smokers also are very, very helpful to these kids, and I'll show you the evidence for that from the peer-reviewed literature. So let's take a look a little bit at these assays real quick, just to familiarize you with them. This is the methylation array approach that's been included through the WHO 2021 aspects of diagnosis for CNS malignancies. There's a couple of papers here from the German group that really began this sort of march toward the use of methylation. And what is DNA methylation? Well, DNA methylation I think about as sort of a signpost. It's like a mile marker along the highway of the DNA on the chromosomes. It basically can signal whether the gene that it's next to or occupying should be copied into RNA and protein, or whether it should be silenced if it's not there, or versus whether it is there. And so methylation is yet another way of either turning on or turning off genes, and is often employed by cancers to turn off specific genes that lead to the development of cancer, or vice versa. So we really need to understand methylation for that reason. The other reason we need to understand it is that methylation marks, those signposts on DNA, occur very early in development as the fetus develops unique tissues in the body, such as the muscles or the brain. And those marks persist. And so in some ways, getting methylation information can tell us about where that tumor is located in the body and what the primary tissue is that it came from. So these are both very important features of diagnosis and can tell us the type of brain tumor. The way that works, sorry, this thing has a mind of its own, I swear I didn't touch it, is that we utilize what's called a CNS classifier. This is basically an algorithm that's been trained with lots and lots of data from lots of previously diagnosed brain cancers, and so when it sees information from a new cancer, it basically can say, oh, I know what this is, it's this. And we can sign that out as a clinically actionable result from clinical testing using this approach. Put the DNA on an array, get the readout, run it through the classifier, you have an answer in many cases. We can also get important information about copy number changes in the DNA, so more than 2 copies or less than 2 copies, and a specific gene that's important in CNS malignancies, which is MGMT, that can tell us about the use of temozolomide or not. So this is an important assay that we offer in our clinical laboratory. How important is it? In this WHO guideline from 2021, look how many changes there were to the new nomenclature and diagnosis of different cancer types. Lots of changes came about by including methylation profiling as a component of diagnosis. So this has been an important development in CNS cancers. I mentioned fusion drivers, 2 genes that come together and make a unique protein that somehow drives the cancer. These are prevalent in pediatric cancer writ large and known in certain brain cancers as well. And so we have an assay from a company called Archer. This is a fairly straightforward assay to run, very sensitive down to small tumor content, about 10%. As you can see here from the bullet points, and really is a targeted assay. It looks specifically for genes that we know form fusions in cancer. So the more we know about cancer fusions, the more genes we can actually add in, and we can be more sensitive to new fusions that have been developed. And I'll tell you that we just recently introduced into our clinical laboratory version 2 of our Archer assay, which included many new genes that have been discovered as drivers in cancer, so that we now have better capability to illustrate that, or to identify those fusions rather. And I'll talk about that in just a minute. Many of you know that there are a variety of NTRK genes, 1, 2, and 3 in particular, that participate in gene fusions, and in some cases do drive brain cancers, and these NTRK fusions are now FDA approved in a very unique clinical trial that involved both pediatric and adult patients, because NTRK fusions are identified in both in cancers of different types. And so this is one example of a fusion driver that is addressable right now today with an FDA-approved therapy. And we have seen kids with these fusions, as I will illustrate. Lastly, I mentioned the sort of comparison of tumor DNA to normal DNA to identify mutations, copy number changes, structural rearrangements, you name it. And this is just a diagram meant to sort of illustrate what we do in our laboratory, which is we combine whole exome sequencing, which just focuses on the coding parts of the genes. Those are the parts that are going to make up that final protein, with a little sprinkle in of essentially whole genome sequencing, which is just looking at everything in the genomic DNA. And this combination gives us two things: a really high sensitivity. We sequence the tumor to a very high depth so that we can understand the makeup of that tumor. Is it a single clone of cells with all the same genome, or is it a mixture of cells with variations on that theme? And we can also look in the normal DNA in this approach for germline susceptibility, and I'll illustrate this for you in a minute. So we identify variants by comparing each DNA data set to the human reference genome, individually, tumor to human reference, normal to human reference. We call all the variants, and then we compare those two to each other. Why? Because the germline variants will be present in both. Sorry, here we go again. But the somatic variants will only be present in the tumor DNA. Okay, and we have to know that distinction, and I'll show you an example of that. But finally, then we interpret variants to identify drug targets, these changes in the chromosome arms that may be prognostic, and germline susceptibility, as well as now tumor mutational burden, because that's becoming important in the setting of using immune checkpoint blockade. Here's just a couple of graphic demonstrations of what DNA data looks like from next generation sequencing. I know you're all excited about this, but I thought it would be helpful to illustrate. Here's a real example of tumor versus normal from one of our patients with a myeloid sarcoma. We identified this very targetable deletion here, sorry again, in a gene called PDGFR beta. This is also a gene involved in brain tumors. And that missing piece of DNA sequence happens right here in the protein. This is the protein that is encoded by that gene. And we know that that mutation actually turns that gene on permanently, starts signaling the cell in an uncontrolled way, and it starts to grow and divide out of control. The good news about this particular mutation is it's targetable by a variety of drugs, dasatinib, imatinib, and this patient received those drugs and remains tumor-free to this day based on that diagnosis. These are just illustrations of whole chromosomes with copy number alterations. Again, it's a pictograph at the top of what the chromosome looks like. Here's an amplification, here's a deletion, here's a whole arm, the P arm of 17 that's been involved. And we also annotate cancer genes underneath of that, so in our clinical sign out, there's information that can be returned there. So what's the role of molecular characterization? I've already mentioned prognosis. These are just a couple of studies in medulloblastomas that came out of COG trials, ACNS 0331 and 0332. The initial trial here was just looking at therapy using different types of radiation, either focused or general whole brain radiation, and illustrated the importance of molecular subtyping, because as you can see, there are different response rates in these different types of medulloblastoma, with group 3 being the poorest response on the Kaplan-Meier curve. And this then rolled into high-risk medulloblastoma approach, looking at different subgroups. You can see the Wnt group here, this group 3 here, and basically the study here was looking at carboplatin versus not, really illustrating that for the Wnt group, probably treatment with carboplatin, not necessary. 100% cure rate based on just surgery here, but not so much for the sonic hedgehog, where it didn't make a difference, significant difference for the group 3 medullos, and this is the way that the standard of care changes. These are great developments, don't get me wrong, but we're still working in the realm of basically a high-dose chemotherapy that is not targeted. Things have changed more recently for low-grade gliomas. As many of you probably know, the setting of treatment with a MET and BRAF inhibitor was just presented at the American Society for Clinical Oncology in 2022. These are just a couple of slides from Eric Buffet's presentation showing that significant improvement in these kids can be obtained with this combination of targeted therapies that, again, have a fairly low side effect profile, are well tolerated, and definitely deliver in terms of improved progression-free survival. More recently, in a different, very rare subtype of brain cancer from the New England Journal and a group in Boston, you can see that BRAF MEK inhibition was also highly effective in papillary craniopharyngiomas. That's even hard to say. Those are very, very rare tumors. But they all carry these BRAF V600E mutations, and they had a very, very high response rate. Again, and admittedly, a very small number of patients, 16 patients, 15 of which had a objective partial response or better with a large reduction in tumor volume and really good progression-free survival. So there is some progress here, and then I'll just finally illustrate the checkpoint blockade in high TMB CNS malignancies. There was a paper in Nature Medicine with a very large cohort, and then a very small cohort, which is an extraordinary paper. I was pleased to be able to review this, and I just wrote the opinion piece about it for Clinical Cancer Research, which is shown here, looking at only 10 patients. But many of whom had remarkable responses in the setting of constitutional mismatch repair defects. And so this is now, I think, going to become a standard aspect of diagnosis, which is to evaluate the tumor mutational burden in these patients. All right. So let's just talk for the last few minutes about molecular profiling and how it impacts care and outcomes. But how do we ensure broad access to full molecular profiling for kids in the United States? This is through the Childhood Cancer Database Molecular Characterization Initiative. Here's a large diagram sort of showing all of the moving parts for the MCI. I'll talk more specifically about the moving part that I'm in charge of, but I thought it might be helpful for you to see how this works. We have eligible patients that now include CNS malignancies, sarcomas, and rare cancers. They go through this characterization program that's being funded by the National Cancer Institute, and we perform a variety of assays, DNA, RNA-seq, methylation, and return of results within 21 days. This is all clinical testing, so it can be used for medical care, it can be used for enrollment onto a clinical trial, and is completely free because NCI is paying for the clinical testing. And we're using advanced approaches to deliver the data out through the Amazon Web Services HIPAA-compliant cloud that we oversee, which is really changing the way that these data are delivered back to providers and the rapidity with which they can receive the information, as well as the rapidity with which we can deposit the data into the childhood cancer database. It's shown down here that this will be a data repository of molecular and linkable data. So the information is completely de-identified, to be clear. There's no PHI that goes across with it when it goes into the CCD. However, there are universal identifiers that allow you to link methylation to fusion to exome to clinical pathology for these patients. What does that mean? It means we're not only helping patients in real time, but we're also perpetuating discovery for the future. And I think that's really critically important. So this is our look at the MCI and how we're performing this molecular profiling. This is a 5-year program. It's being done in collaboration with the Children's Oncology Group, or COG, which is the largest NCI-funded cooperative group in the United States, but also includes sites in Canada, Australia, and New Zealand as well. We're intaking the material in a unique way. It's all digital. No pieces of paper come over with the samples. Just digital information gets pulled over when they come across the street from our biopathology center, which is the banking biorepository for COG. So that was already in place. And they are also at Nationwide Children's Hospital. We perform the laboratory tests that I mentioned, the bioinformatic process, analysis and interpretation, and sign out of a clinical report, which is then delivered using our Amazon Web Services data delivery. We also crunch all of our data in the bioinformatic processing step in the Amazon cloud so that we can take access of as many CPUs as we need for all the patients coming through, which I'll show you in just a minute. And we also accomplish the data deposition into the childhood cancer database that way. You'll see here that we're required to deposit it in 90 days. We joke that we actually get it there in about 90 seconds because the processes automatically redact all the PHI, so there are no mistakes with redaction, and send it through the Amazon Web Services cloud, which is very, very rapid. The really good news here is not only are we doing things rapidly, but all of the folks at the NCI that are responsible for posting data into the childhood cancer database are getting the data there in a very rapid way, and there are over 1,200 patients already worth of data that have been deposited there, so we're pretty excited about that. Here are just some other facts and a few figures for those of you who might like to look at them. One on the top is just sort of the ramp up since March of 22 when we had our very first CNS malignancy patient enrolled from Seattle Children's, if you're interested. And this has been growing quite rapidly. As you'll see here, we're going to scale up to 3,000 cases per year during this coming calendar year, I should say. And that will be accomplished by including in neuroblastoma and Ewing sarcoma patients, as well as the patients that we're already studying, which are CNS malignancies, soft tissue sarcomas, and a group of rare cancers that I don't have time to list out, and I couldn't if you really asked me, but that information is available. We already have over 45 states worth of patients enrolled, and as I mentioned, tests already conducted and returned on patients from Canada, Australia, and New Zealand. Data already available at the CCD. And COG is also collecting information on whether the MCI changed the diagnosis for the patient or changed the treatment. And they're getting this information back from the enrolling oncologist. So we'll kind of have a complete picture, if you will, about the impact that this has had on kids with cancer. Here's just an infographic of how we keep track of our progress. We have currently enrollment at about over 2,000 patients into MCI. We have about 1,800 in our system so far. And here's a great result, I think. Over 90% of patients receive at least one cancer-relevant result from the clinical testing that we perform. And so I think this is going well. This is data current as of beginning of September. We keep this updated monthly, and we have monthly calls, and I'll have one later this afternoon. If you see me disappear, I apologize. That's where I'll be. So I wanted to spend a little bit of time talking about a few of the aspects that I teased out of molecular profiling, just to give you some more facts and figures. What about germline susceptibility? I mentioned that, and the estimates vary in terms of how prevalent this is in pediatric cancer. So far, based on the tumor types we've looked at, about 14% of patients have a pathogenic or known pathogenic. So these are not guesses, these are particular mutations in particular genes that are known to drive cancer development from the germline. And you can see the total cases and number percentages from both copy number CNVs or from point mutations. That's what SNV, single nucleotide variant, stands for. But in total, in pediatric CNS malignancies, we're at about 14% overall, much higher for rare tumor types as shown here. If you're interested, here are just some examples from what we're finding in particular mutations and the genes that are involved, P53, CHEK2, et cetera, and the proportionality there. And then specific copy number findings are listed here, along with the cancer predisposition syndrome that's associated with them. And these are sort of onesie-twosie events at this point in time, but in many cases are sort of cataloging things that we haven't really been able to identify in the past. Why? Because often the germline isn't tested, so we have no sensitivity to detect these things. I don't think that's right. The second reason is because many tests that are conducted are looking at a very, very small number of genes, a panel as it's commonly referred to. We're looking at every protein coding gene known in the genome, the exome, if you like terms. And this, I think, will perpetuate that kind of discovery future forward, because if you only look at the genes you know about, you'll never learn anything about the genes you didn't look at. And so I think, sometimes people are like, well, why are you testing all those genes? We really only care about a few. In fact, we don't know about the ones that we don't care about yet. Maybe we will find that out pretty soon. Here's just a look at CNS fusions. Many of these are prevalent in low-grade tumors like this Kia-BRAF fusion, but you can also see some NTRK fusions here, EGFR, which is highly targetable, PDGFR alpha, which is the beta homolog, etc. So there are a number of targetable entities here. FGFR, internal tandem duplications, are targetable with therapies that are FDA approved and available. Let's take a quick look at a case example just to finish things up. I thought you might like to look at the data for just one patient, a very intriguing patient, 10-year-old female with a high-grade brain tumor resected from the posterior fossa. So this is the Archer result for this patient. Nothing was detected for fusion drivers for this patient, okay? So this is a signed out result that says nothing detected. Keep that in mind. Here's a methylation report that we got for this patient, and the methylation report was a very high score, almost 1. CNS high-grade neuroepithelial tumor with MN1 alteration. Keep that in mind. And you can see here some of the copy number data. This is chromosome 1 all the way to chromosomes X and Y. So we're looking at every chromosome here, and you can see a variety of different copy numbers that have changed, mostly deletions, in the red. And in particular, this looks a little bit messy over here. We'll get back to that in a minute because we have better resolution from the DNA comparison tumor normal exome. Here's the germline analysis. This patient has a known pathogenic mutation in a gene called ATM. This is a known cancer driver. And the copy number in the germline, the normal DNA, looks very quiet. There's just that single line across all the chromosomes from 1 over there on the left to X and Y over here on the right. So normal copy number profile. However, when we get to the point mutations and the copy number for the somatic tissue, for the brain cancer, there are no somatic cancer-associated point mutations, but lots going on that we've identified on chromosome 22 and chromosome X, and here are all the genes that are involved. And you see this big word that's almost impossible for me to pronounce. It's called chromothripsis. That's a fancy way to say that those 2 chromosomes have been highly altered by copy number changes. How about that? So let's take a closer look. This is just the screen out for all of the chromosomes. Again, this is now from copy number out of the tumor normal exome. You can see these other changes, but the really messed up pattern here. And now we can see that in close-up. Here's chromosome 22 on the top, and here's chromosome X on the bottom. And things are kind of all over the place with those different deviations from the line. That's how we call chromothripsis. But importantly, there are a couple of genes that are involved here on these 2 different chromosomes. One is a gene called BEND2 on the left and MN1. Remember that gene? That came from the methylation analysis. Well, this information tells us that there's probably been a fusion between MN1 and BEND2. Now, for those of you who are really paying attention, and I'm sure that's everyone, you're like, wait a minute, Elaine, you told us there weren't any fusions, so what the heck is going on? Well, this is an example of a gene that we just added to our fusion panel, MN1. It wasn't there on version 1, and this patient was tested with the version 1 fusion panel. This is the value of doing multi-omics as well as all the other values that I mentioned earlier. We often can integrate this information and find things that we might have missed that were indicated by one test, methylation, missed by one test, fusion, and picked up by another test. And we can actually go in and look specifically at the individual reads for this patient, like I showed you earlier on that PDGFR beta deletion, and we can find evidence that that fusion has occurred. I won't bore you with the details of how that's done, but it can be done. So this is important. This now results in a very precise diagnosis for this patient. That combination of information plus pathology tells us that this is an astroblastoma that is MN1 altered, and there is new information about genetic susceptibility because there wasn't any before. This patient had an ATM pathogenic mutation, as I talked about. So this is just an example of what we can do from a single patient using the MCI approach. I am going to finish now with 3 last slides to talk about challenges because we're not done here and we aren't anywhere close to being done. So what are the scientific challenges and what are the medical challenges? One scientific challenge that you all are involved in and can help out with more is we need to incentivize drug development for pediatric-specific drivers that encourages novel approaches to drug design, because not all of these are simple tyrosine kinases. We know how to drug those, but we need PROTACs, degraders, and other terms that I don't really understand. And we need our medicinal chemists and chemical biologists in academia to start thinking about what that's going to take to really address pediatric specific, not that convenient crossover with adult tumors. We need to devise better model systems for pediatric cancers because mice are not suitable model organisms for a variety of reasons. And one of the primary reasons is it's very hard to recreate pediatric cancers in mice. I'm not a mouse modeler, but this is what all of my mouse modeler buddies tell me, so I guess I have to believe them. But there are new approaches such as screening disrupted cells from a patient's tumor that are now emerging. If we only had more drugs to look at, that would probably be a better equation. There are also a way to make tumoroids or tumor organoids for testing and for CRISPR engineering, et cetera. So better model systems are important. Here's a real campaign that I'm on right now in my own lab, and I think more people should be involved in this. We don't really understand normal brain development. We understand fetal brain development. We understand what adult brains are like. They're pretty boring, as it turns out, from the standpoint of gene expression. But we don't really understand that incredible time from postnatal to adolescence in terms of all the crazy things that go on in all of normal brain development and in all the tissues in the brain. We just don't. And that's really important to provide a baseline for understanding pediatric brain cancers, which are abnormal. You can't understand abnormal until you have a normal comparator. That's all there is to it. And we really need to get to that. We need to get to that in terms of RNA as well as methylation profiling, and also looking at the way the DNA is ranged in the cell, in the normal cell. That's called chromatin profiling. These are all very doable, but we haven't done it yet. We need to look at different ages, age groups, 0 to 3, 4 to 12, 13 to 15, because we know the brain changes in adolescence, right? Those of you who have adolescent children or have had them like me, you know, it's very different going through puberty and things change. And we even know that in the low-grade glioma setting, the brain tumors quiet down often in puberty. Wow, if we understood how that worked, that might be actually impactful for other types of tumors. And here's the big one in the room, the elephant in the room. Men are different than women. Boys are different than girls. This is a revelation to you, right? But it's very true in terms of brain development as well. And so we really need to understand both male and female normal brain development to get that perspective. And finally, again, obviously, the pediatric immune system is much different in adults, and we need to develop therapies that are suitable for kids. Immune checkpoint blockade therapy is a great thing for adults and now for some small subset of children. But I will also tell you that one of the realities from those papers that I mentioned is that kids with constitutional mismatch repair defects also have the most severe side effect profiles from immune therapies. So it's not a perfect solution in some cases. And then medicine needs to change. We need to better educate our oncology fellows, our pathology fellows in the pediatric setting, and even our surgeons to some extent in terms of what is genomics, what is molecular profiling, how is this information even used? Depending upon when you finished your fellowship in medical school, you may not even understand what the genome is, maybe not even as well as you guys do from that very first slide that I had. So we really have an obligation, not you guys, but people like me, to train fellows and residents around what genomics is and what it can do for us. In rare poor outcome cancers, we really need to push frontline trials of novel therapeutics. I'll get to that on my last slide coming up. And we have to continue to share information. One of the great things about changing into a pediatric setting in cancer research is that we really do share information very, very well in terms of not trying to keep it as an institution-specific, gem that we can't share with anybody else. We have to do this in the setting of rare cancers. But it would also be nice to really encourage case reporting, those individual patients like I just showed you, and what we learned from them. And give physicians CME credit or publication credit because they need this sort of thing for tenure and progression in their careers. It's hard to put together in a publication, and it's hard to get publications published. Trust me, I know that. If there was a way to sort of do that in a more facile way, I think that would be really encouraging and would really encourage this critical aspect of data sharing, and ultimately will really lead to what I would call artificial intelligence augmented treatment planning, because we can use artificial intelligence to glean information from all of these case reports and start to develop information about how to treat that next patient that I think will be really informative in the future. And then just one last thing in terms of how to change the profile of clinical trials, because it's hard to accrue enough patients in a reasonable time period, especially in a very rare disease type, to get information and change the standard of care. So one of the things that we're doing at our hospital is we are sponsoring the Connect Consortium, a collaborative network for neuro-oncology clinical trials. It's hosted at our hospital, but as you can see, there are multiple performance sites across the world and the United States. We have to turn cancer clinical trials into something that is worldwide. And there are other countries that are doing this, and I think it's critically important for that time frame to shrink and for these drugs to get approved as they come along. And this is just sort of the future. This is what MCI is going to be doing for the Connect trial that's beginning to open this year and will continue opening arms through the next year. You can see different types of alterations that are being identified here through the MCI combination of assays, and we'll be able to assign kids with high-grade gliomas, DIPGs, DMGs, and the like onto these individual treatment arms, combining specific targeted therapies with radiation therapy in the first line. I think we have to do this to start to move the needle on these very difficult diseases, and I'm really excited to be a part of the Connect Consortium. So that's all I have for you today. Thanks for your attention, and I'm happy to answer any questions now or later if you're shy. Yes?
Hi, with the methylation profiling, is that something that's offered to every child with newly diagnosed? Is it something that's cost-effective for the hospital, or is it something that is covered by insurance? Is this done for all patients?
That's a great question. I wish I could say that it is. It is part of the standard of diagnosis, as I mentioned, in the pediatric setting. So not all hospitals offer it. It has to be a CLIA-approved test through CAP-CLIA in New York State if you're a hospital in New York. However, there are ways to transition DNA from a biopsy to the National Cancer Institute. Ken Eldape, who's a very eminent neuropathologist there, will accept in samples for methylation profiling and sign out clinically. And there are other hospitals that will offer that as well. Children's Hospital of Los Angeles does. We do. I'm pretty sure CHOP probably offers this, although I don't know that for sure. Maybe Adam can address that. And then New York University as well will provide that. So there are ways to get it done. That's typically done at the NIH, I know, through LDAPE service at no charge to the patient. And the reason why is they do want to keep the data and continue to build and improve that machine learning predictor that I mentioned, that algorithm. And I think they really could use a lot of pediatric samples actually as well. So that's a discussion to have with the oncologist or pathologist about submitting that sample. The turnaround time is pretty good. It's probably somewhere on the order of maybe 4 weeks. I don't know exactly what they provide. And, you know, they get relatively more or less inundated depending upon what day of the week it is. So I think there is a promised turnaround time because we have to do that as part of clinical testing. But that's one way to get it done. I do believe it is reasonably well reimbursed by insurance. It's not a super expensive test because it's not sequencing, it's just an array. But because it is part of the standard diagnosis for CNS malignancies only, you would be able, as a hospital lab, to get reimbursed for doing it, and I know that we do. I don't know the level of reimbursement or how well it compares to reality, but often children's hospitals find ways to pay for the overage. I know ours does through a variety of state and U.S. government programs. So you should be able to get that test one way or the other. Mm-hmm. Yeah.
Thank you. That was a great talk.
Thank you. Good.
I'm wondering what you can do right now with liquid biopsy, if anything.
Yep.
And then also, sorry, I'll just ask that question again now that I have the mic.
Sure.
Wondering what you can do with liquid biopsy right now. So instead of having to take a tissue sample, just using the blood or CSF.
Mm-hmm.
And also, is any of that included in the MCI?
Great questions, thanks. Liquid biopsy, for those of you who don't know, is sort of a way of sampling the tumor based on the fact that tumors often release dead cells into the circulation, and so we can find DNA, RNA there that's from the tumor. It's obviously in the mix of other normal cells that are also dying and, you know, pushing their DNA and RNA into the circulation. The answer to your second question is not currently a part of MCI, nor is it a part of the standard of care for pediatric patients, unfortunately. We're working to try and change that, and I know others are too. I didn't have time to mention it, but on that last slide that I showed with the many, many arms of the umbrella trial that we're opening through Connect, the big green box at the side said something about it. And basically, the way to change the reality in that it's not being performed now in most settings for pediatric patients is to link performing it in the research setting to a clinical trial like the Connect Target study. And so what we will be doing as that study rolls out is we will be collecting CSF, cerebral spinal fluid, and blood from patients longitudinally before or as they're diagnosed, and then over time as they come back into clinic for imaging or what have you. And we will run correlative evaluation because we know what their tumor profile is. So that will be happening, and we hope to use that to sort of help add to the evidence that this can be really important for understanding response to therapy or emerging resistance. Those are sort of the two mechanisms by which we look at liquid biopsy. The other thing that I think is going to be really impactful is in the setting of cancer susceptibility or patients that have already survived cancer. Why? Because I think we will be able, and there are now, I just learned of a study that will be published soon in Cancer Discovery from a group in Toronto in the pediatric setting as well as the adult setting for adults with cancer susceptibility like BRCA, et cetera, where they actually profiled a number of patients over time using blood-based monitoring, not CSF in this particular case. In that study, they were able to show that using liquid biopsy in the setting of a person who is going to develop cancer or has already developed cancer, you can detect a new occult cancer, occult to imaging, 18 months on average in advance before it can be detected by imaging. So I tease my radiology friends that they have got some work to do in terms of coming up with better and more sensitive imaging. But the bottom line is, and we're planning to introduce a study like this in our hospital, for hospitals with large cancer susceptibility and cancer predisposition clinics, we need to start evaluating these kids when they come in for their once-a-year whole-body MRI and get a tube of blood and start, evaluating when we can pick up these signals as a means of moving the needle in a really important setting, I think. So thanks for that question. Yes?
So, if you don't mind, I want to take it back up to like 10,000 foot from a genomic perspective.
Sure.
So when I look at this now, and I'm a physician, when I look at this and I start to think from a prognostic perspective, from a predictive perspective, from a preventative perspective, all the way through therapeutics, that what really, it seems to me, needs to happen is that for us to start, as we're doing, collecting this data now, which is not that simple, with the idea that over time, not only will we be able to predict and prevent, but when you are diagnosed, we'll have a database of information of who has this genomic and phenomic presentation, and this is the right treatment based on that database. Is that correct assessment?
It is to a first approximation. I think in general it's correct. I do realize that as much as we'd like to categorize every patient into a neat box based on their methylation, tumor normal exome, and fusion analysis, there are always patients that are outside the box. So generally speaking, yes. This is why case reports are so important. 5 years ago, I started a journal out of Cold Spring Harbor Press called Molecular Case Studies at a time when nobody else was willing to accept case reports, with the exception of the New England, who does take, exceptional case reports that are potentially practice-changing. Now, today, there are literally everybody has, precision oncology, accepting case reports or small numbers of patients. So I do feel like that continues to be important. But yeah, absolutely. And I'm sure Adam will talk to this as well, because he's put together one of the largest databases already in pediatric cancer across the board. I think it's going to become incredibly important. Right now, today, we have at our institution a faculty within the Institute for Genomic Medicine, a woman who trained at Baylor Texas Children's, and I refer to her as the genome whisperer. Why? Because when there are oncologists within MCI that get results they don't understand or can't interpret, I send them to Samara Potter. When there are oncologists within our own hospital that don't understand what to do with the information that we give them, I send them to Samara Potter. But Samara is just one woman, and she's amazing, but we have to, develop this capability as a query service that any oncologist can tap into with the data that comes out of molecular profiling. That's, I think, the longest term goal that we have in mind. With, let's face it, 20,000 patients maybe at the end of the MCI worth of data across a multitude of pediatric cancers, maybe we'll get there. I think it's a good beginning. I think it will combine with other databases like you'll hear about over the next couple of days to really, move the needle significantly in that important direction. Sorry, we have to wrap. So sorry, if you have other questions, just feel free to come up. I'm happy to discuss. Thanks so much.
All right, everybody, we have a break actually we don't have a break, but we're gonna switch gears to two different track sessions. So look at the schedule, decide which room you wanna be in, and we'll start as quickly as possible since we are running a little bit behind already.
Is there a particular order that you need us?
Good afternoon. Thank you so much for joining our panel on intention and advocacy. If you could just take a seat and we'll start our discussion. For those of you that don't know, I am the Director of Advocacy for the Pediatric Brain Tumor Foundation. My name is Michael Henry. And on this stage, I'm joined by some of our great advocacy partners. Every person on this stage represents some of the incredible work that our community is doing. I just wanted to give you some background on why we decided to talk about intention and advocacy. We thought, what's the best way to equip this community with usable, strategic advice moving forward? And we were thinking about how do we help folks move away from reactionary work into proactive work, and planning out our work in advocacy so that we are moving towards a goal and with a specific intention in mind. And again, every single person on this stage is a great example of that and how they've worked over the years. So I'm just gonna ask everyone to maybe make a brief introduction, who you are, who you work for, and maybe your backstory.
I'll snag the mic from you. There, better? All right. Hi, everyone. I'm Sarah Milberg. I'm the Director of Government Relations and Advocacy with the St. Baldrick's Foundation, and I also co-chair the Alliance for Childhood Cancer. And I got here by way of kind of government affairs work and also a little bit of a personal connection. My husband is a young adult survivor who has been cancer-free for 11 years.
I'm afraid to move this now. Danielle Leach, I am the Chief of Government Relations and Advocacy at the National Brain Tumor Society, co-chair emeritus of the Alliance for Childhood Cancer. I am a sibling of a childhood cancer survivor of rhabdomyosarcoma, and my son Mason died of medulloblastoma at age 5.
Hi, I'm Dean Crow with the Raleigh Foundation for Childhood Cancer Research and a member of the Alliance. Mm-hmm. And I got here because my husband coached the baseball team and one of his players got a brain tumor. So that's how we got involved.
Hello, my name is Ellen Miller. My daughter, Gabriella, had DIPG and died actually coming up on 10 years ago. And we started a foundation called Smashing Walnuts. It's evolved where we are now working on advocacy, and I am working to pass a piece of legislation that has her name on it that funds childhood cancer and disease research.
Great, thank you so much. I should mention too, I'm Blair's dad. I lost my daughter to an ETMR about 3 years ago. And like so many of you, she drives a lot of what I do in the advocacy space. I'd like to just start off with a question for everyone on the panel. There's so many different approaches to advocacy. How do you personally approach advocacy, and maybe how has that changed since you first started? If anyone wants to go.
Sure, glad to kick that one off. So I think the way I approach it first is by figuring out what my goals are. So do I want to secure more?
Yikes.
Okay. All right. Am I trying? All right, I'm just going to be very loud. Perfect. What are my goals? Are my goals to secure more funding? Is it to get a new program implemented? Or is there a certain challenge I'm trying to overcome? So it tends to be where I start. And then I think about what do I need to get there. And typically for me, I'm very detail-oriented, but also a little bit data-driven. And so I tend to start first with what information do I need to get the right message across. I think being accurate and being a reliable source of information is really valuable in advocacy. And so I think I know just enough to be dangerous in a lot of spaces, but I also try to know what I don't know, and then I try to find out. So that tends to be how I start. And then it's typically, especially because the community is really small but mighty, is who can I bring in to help, and how can we work together in a collaborative way to get something done, but all with that top-level goal in mind.
Similar to Sarah, you start off with the goal, but I always say if you're not at the table, you're on the menu. So making sure that the childhood cancer community is at the table at every opportunity that they possibly could be, and not just in Congress. And I think that that's something that we often don't think about when we talk about advocacy. Everybody assumes we mean only about Congress, and there are plenty of opportunities in your local hospital, with pharma companies, with advisory groups, with government agencies we have advocates all over the place, making sure that we are at the table. So often in my process, I stop and see what exists. Well, who is working on it? How can I collaborate with them? Also, to your point, advocacy and making sure that the facts are correct. Also, really understanding the opposition. What's the opposition to something? Why are people opposed to it? Or what are the underlying circumstances that I may not understand? Because often on the surface, it sounds like a great idea. Your one-pager is often just a brief summary of a piece of legislation or an advocacy alert. And to get a more fundamental understanding, in the reactive space is really trying to make sure that we understand why somebody may not like it is just as important as why we want to support it. And then my advocacy is mostly focused on educating and engaging advocates and building champions on the Hill and building relationships on the Hill and making sure that advocates feel empowered to build those relationships for the long term, so that as we have specific asks in advocacy for the childhood cancer community and the pediatric brain tumor community and brain tumor community overall, we have those champions that are ready, that understand our issues, and are ready to work with us as we move the needle.
Very good. I agree with everything that they have been saying. So just to expand on something a little bit different, I think it's really important when you go to advocate that the message that you're advocating is very understandable. You're pretty much going to be advocating to staff members if you're doing, and most of our advocacy work is done here on the Hill, and you're going to have a 15-minute meeting, and you're going to have to make sure that you can explain what you need and want and why. And I completely agree, and I know Ellen does too. You have got to have the facts and your data have to be correct. So that is super important. But a message that they can understand within that 15-minute meeting, you've got to kind of make it simple enough for them to understand, and then leave behind your white paper or your one-sheeter that has that data to back that up. I think that's super important. It's also really important to know what else is going on in the childhood cancer advocacy space. so that you are not overstepping or asking members to do the exact same thing, which is really frustrating for the members and also embarrassing for your member, you know, who's going to be backing you with what you're doing. I also approach it, and this is the truth, the staffer is your best friend. Everyone wants to get to the member. Everyone's like, no, I need to meet with the congressman. You really, I mean, you want to meet them, yes, but your meeting and moving what you want to have happen forward is through the staffers. So staffers need to become your best friend, and you need to be respectful of the 15 minutes, and you need to be sure that, again, that your message is clear, what you want and why you want it, and do it within the 15 minutes. So I would recommend that you practice.
So just going off what you just said, you know, we of course want to meet with the members. The members like the head, but the staffers are the neck that moves the head. So that's really important to know that our staffers, you know, if you insult them, they can ruin your advocacy. So first, I actually want to ask everybody, show of hands, who's done advocacy already? Okay, take a look around. Okay, that's great. I love that. And I love also seeing that not everybody has. And so I feel like I'm very much like you all, because I never did any advocacy. I would like sign something that had come across through an email, but I had never done anything until, Gabriella came along. So 2 weeks before Gabriella died, she was interviewed. It was sequestration 10 years ago, so government shut down because they can't balance their budget. We knew kids that were going to the NIH and NCI campuses for their treatments, and they were being turned away because the government was shut down. And the interviewer asked Gabriella if she could say anything to our elected officials. What would she say? And she said, stop talking, start doing. And that video clip caught the attention of former House Majority Leader Eric Cantor. This was not my vision. I never saw myself 10 years ago being up on a stage with these amazing advocates doing this. I was you. I was in the audience. And Gabrielle had a different plan for me. And so that led to her name being added to a piece of legislation. So you asked about goals, and you could hear from all of these amazing women that, you know, the goals are different because of who they are and what they're doing and who they're working with. So for me, my goal is I'm working on getting a piece of legislation that I'm working on that has Gabriella's name on it, passed, that will fund childhood cancer and disease research. So that's my immediate goal. But goals, they're not quite so, you know, singularly focused, because as we work with other people, we're helping them with theirs as well, and they help us with ours. And so as folks that are interested in becoming involved in advocacy, you don't need to have a yearly plan. I mean, that could be intimidating and you're like, I'm out. Don't do that. We need you here because your voice, your story is so important, and we all are the ones that move the needle. So reach out to me, reach out to all of these ladies that are on this panel, because they've got things that help our community as a whole. And you know, you might not like mine, but you might like somebody else's. So definitely make a point of doing a little bit of research and taking up what it is that you can bite off and help us. That's very much appreciated.
Got it. Thank you. One of the things I think all of you touched on was relationship building. I want to circle back to that, but first, I'd just like to hear your advice or your opinions on, there are so many different opportunities in advocacy that I think all of our organizations present. How do you personally narrow down your organization's focus, and what advice do you give to advocates who come to you and they don't really know where to start or what to focus on, what initiative to focus on? How do you start that conversation with them?
Just to kick that off, I think for me, being involved with the St. Baldrick's Foundation, St. Baldrick's has a very clear research-driven mission, so that tends to be, when I think about what to focus on in the advocacy space, I think I go back to our mission of, That does not like me. But the goal is always to advance research. And so if we're trying to secure federal funding for research, that fits nicely with our mission. So that is very much my lane. I'm going to try and stay in it and bring some friends along to do it. So it tends to be where I start. I also, to the points that have been made as well, think about what other people are working on. So, for example, I know Ellen's working on getting the Gabriella Miller program reauthorized. That's a key priority. That has to happen like now. And so while I may have a certain policy goal in mind that's more general or more broad, that's an immediate need. So I may turn my attention to help my fellow community member to make sure that the policies that support the broader community are happening in the right cadence. And that goes back to doing your research, knowing what some of the kind of busy times and key deadlines are, knowing, even just looking at the congressional calendar, which is available online, and it's never exactly as true as what's printed at the start of the year, but we try. But there are certain things we always know to be true, right? Like the fiscal year, for the federal fiscal year, always ends on September 30th, and I think this year being a little bit tougher than typical, but we always know this time of year there's going to be an opportunity to kind of put your thumb on the scale a little bit as budget discussions are happening. We know in March, as the budget process is ramping up again right after the State of the Union address, that's another opportunity. So thinking about when things are busy, when some of those ebbs and flows are, that certainly helps. And then just making sure that I'm staying in the lane that I've chosen to be in just by following the mission of my organization.
As an organization, I'll put on my organization hat and then as a parent hat in a moment. So as an organization, we analyze what potential policies are going to be moving in the next Congress every single time at an annual basis. So we reassess what's moving, who's working on what, we track everything, and then we decide, okay, we know that research funding and access to care are incredibly important to us. So those are and drug development. So those are our three main buckets. And then there are policies under each of those and opportunities for appropriations that we focus upon, like the DoD peer review cancer research program, making sure pediatric brain tumors and brain cancer are eligible categories every year. So there are specific things that we work on with under that umbrella that we take the spearhead efforts on, but then other times we participate in coalitions like the Alliance for Childhood Cancer and others that allow us to monitor and track legislation that allows us to engage and support work on drug shortages, other opportunities for engagement around access, that we wouldn't necessarily have the staff. We're a staff of 3, and it's not huge. And we also serve the adult and pediatric space. So, and there are many dual opportunities. There's more commonality than not in the brain tumor space between the adult and pediatric in terms of opportunities for engagement and advocacy. As a parent of a child who passed from a brain tumor, what I can say to you is join the action networks. You don't have to jump in and go to Congress the first week. And everybody, depending on what stage you are in your brain tumor experience, your advocacy is going to look very different, perhaps from the beginning all the way through the end. There are some people who jump in both feet from the moment of diagnosis, and they're up on the Hill, and they come to action days, or they come to Head to the Hill, and they're on it. And there are other people that they're not ready for that. So I think thinking about the opportunities in the digital space, many of us have action alerts and ask you to jump in, often follow our social medias, because often we'll put out calls to action or calls for people like, hey, we need somebody from New York in, I used to work for Nita Lowey's district, and we're always looking for people who have specific stories so that we can then build that champion in that office. So that's my recommendation, is at least do that, because then you know what's going on, and then you can decide where you are in your journey and what you're ready for at any time.
Completely agree with everything that these ladies have said. I think the focus, when Rally kind of found its focus in this space, someone had asked us, where can we get more federal funding? So there was a big battle cry within the childhood cancer community several years ago, more than 4. Our kids deserve more than 4% of the National Institutes of Health budget, which was so true. I want you to know your voices were heard, and now it is 8%. It is 8% that is being gotten, and that is great. Of course, now I want more than 8, I'm just to be honest, but I do. But then I was asked, where can we get additional federal funding outside of the NIH? And I was kind of assigned to really look into getting funding from the Department of Defense Medical Research Program. So that really became Rally's focus, and really had to dig in and learn about that. And that is something that you have to go back every single year and advocate for that funding to retain the report language so that it will be in there. Cancers in children, adolescents, and young adults, the pediatric brain tumors, neuroblastoma, all the different diseases. So since we started advocating, 6 years ago, we've gotten $144 million from the Department of Defense. So, and that has really helped by your voices, but our advocacy work is really done on a committee level, a subcommittee. So really, we are always looking for people who live in the district of the members on the subcommittee. The other focus that Rally always has is to be collaborative and helpful however we can be to someone else's legislation, or to sign on to someone's letter, or to, help them get, there's a piece of legislation that's being worked on for Medicare and Medicaid kids, and they needed a kid, who could tell a story, who couldn't get care by crossing state lines. And they called Rally and said, can you help us? This was another organization. We were like, absolutely, and we got them a child. So I think, being willing to collaborate and be helpful however you can is really, really important. And that is Rally's focus. So our focus is definitely the DoD. We work it probably 365 days a year in all kinds of different ways, because it's a full circle type thing that you have to work on, from consumer, reviewers all the way to continuing to fill out the bazillion forms that they want you to fill out. Yes. That's so much fun. Mm-hmm. And meeting with the staffers is really important. But then also just helping people with their other initiatives, and helping to be a voice for them too is really important. All of our voices together saying the same thing, we are so much stronger and we move the needle better.
Okay, so what a goal of everybody should be in this room, very simple, this will help you focus, is go on to the Alliance, and go on to St. Baldrick's and MBTS and Rally, and they've got a newsletter that you could sign up for, and do that, because they've all said a lot of information, and that's like, oh my God, that's a little bit overwhelming, but it doesn't have to be. So what you can do is educate yourself. You'll get an email. If you're in the mind frame to read it, do. If you're having a bad day, delete it. It's okay, you'll get more.
You'll get more.
But it educates you. And then you're learning about things that you didn't know that are important to you. They're helping to educate you on that. So, so that will be a goal that will help you focus. And so with regards to focus, like again, I'm an individual, I'm not with an organization, I'm not getting paid, I'm doing this, again, as we all are, to honor our children, right? So starting out, it was really intimidating. Like, I didn't know what to do. I made a lot of mistakes. It's okay. I was on calls with staffers or members or in front of them, and I would tell my story and become a hot mess, crying. And at first I was like, I'm embarrassed. And I was like, no, because you know what? We love our kids. It's the worst thing in the world to have your child pass away, and showing emotion, we're powerful as parents and siblings. Show the emotion. It's much more powerful than having a paid lobbyist come in and talk with them. So what I'll also, actually let me ask folks, like, who here has a foundation or an organization that they work with? And do you all do events? Okay, so if you do, make this a goal and a focus, because this will help you with advocacy. As you have events, invite your elected officials on your local level, your county, on your state level, and federal level. They love that because it's a platform for them, right? It's a photo opportunity for them. But that benefits us. I mean, let them do that, because that benefits us when we want to go and reach out to them. So the members will bring a staffer, your new best friend. Doesn't matter if you voted for them or against them, you love them, for however long they're in office. So by doing that, you're creating a relationship, but on your terms, like it's your event, right? And then when you want to reach out for and ask for a piece of legislation, you've already got that in. And so that, I would say, is start by creating those relationships. That's something you can do on your time. And those are things that they carry over for forever.
And as a former district staffer that used to schedule for a member of Congress, I will tell you that that is an incredible tool to build a relationship with the member of Congress, and it's very, very helpful for the long term. And if you get your district staff or this locally based staff on board with you, it is incredibly helpful as you move towards potentially asking them around legislation.
So to that point, we had an event, and we invited just our local, our state, and federal members. And my current congresswoman was just our state level. And then when she went to Congress, now she's the author of the 2.0 version. So that was because I built, she went from state to federal. Did I say that wrong?
No,
So, okay.
but that's cool. That's
And
just
so
how it works.
that's because I worked on building a relationship.
One thing, I do want to say, we are incredibly fortunate in this community that we are a powerful force, and that there has been a lot of collaboration, not only on this table, but outside of this table, working with we have two big coalitions, the Alliance for Childhood Cancer and the Coalition Against Childhood Cancer. I know Sarah kept up the tradition of talking, once a week or once every 2 weeks amongst the coalitions. It's all about communication and talking. When we talk about the peer review cancer research program, we talk and we're like, okay, who did you talk to? Okay, we've got the letter going. You have the letter going. It's making sure that we're communicating with one another, and that's a critical point that we need to make sure that whatever you're doing, to go back to your point before, because you may not know that the coalition has a huge ask coming, or we've been talking about a specific thing with members of Congress for those organizations that have staff or the coalitions that are working on the Hill, and you may come in and have a great idea, and it's great, but then they'll go, wait a second, but they're saying this is the priority, and it confuses them. And Congress has thousands of decisions to make a day. They have thousands of people coming to them. They go through, the staffers that you meet sometimes more than 20 times with different disease groups. Everybody's issue is important and feels important, and those are their constituents that they're serving. It's all about communication and follow-up. And that is incredibly important when you do your advocacy, is about follow-up.
Can I say one thing, Mike? To your point about inviting your elected officials to your events, when you have that is so important, because who's going to be at your event are going to be probably people who can vote for them, and they want your vote, and they want to be in front of you and your people who are in their district or in the state, or depending on if you're a senator, congressman, whatever it is that you're doing. So you inviting them to the local events is really so much more powerful than you can imagine, because they want to keep their job, whether they're the state level or the federal level. And being at your event allows them to work the crowd, and then it gives you the opportunity to tell them what's super important about childhood cancer.
To that point, really quickly, if you are staunchly on one side of the aisle, and your elected official that you voted for is not taking up your cause, maybe you need to rethink who you vote for.
That's a good point. I think just to kind of sum up what a lot of you have said is your message and your communications are much more likely to land if you've been in constant communication with your community, staffers, legislators, other stakeholders. You have to build up that trust, I think, over time, and that's the important thing about collaboration, and all of us collaborate here at this table. just to kind of drill down into that relationship building, can you just talk about how, maybe give an example of how you've built a relationship with either a staffer or a coalition partner, and how that relationship eventually paid off for you? And I'm specifically looking at Ellen, because you had to build those relationships in order to get your bill first passed. And I know those relationships maybe have evolved since it was first passed, and now that we're trying to get it reauthorized. Can you just talk about how that dynamic has changed?
Yeah. Well, so I actually kind of laugh to think that I'm up here on this stage with these amazing people. Like, how did I get here? The fact that I'm thought highly enough is because of the relationships that I've built. I've worked with all of these people up here. I share with them. I've attended their meetings virtually that they have with their staff about what's going on, sharing our legislation with them. And I'll tell you, sometimes I've not been happy with them because I'm like, share my information. And they're like, but you don't have it all lined up right. And they're right. I mean, I'm learning, like, you have to crawl before you walk, before you run. And so because of them and saying, you know, we need this. Okay, now I know I need to go ahead and do that. And now I can send a text or an email, and we respond right away. And I know that if I need help with something, they're there for me. And so in the last Congress, what had happened was, thanks to these folks up here, and thanks to all of you, last Congress, the Kids First passed the House, and then Congress is closing. Like, our elected officials have one foot out the door, and they're doing all of their last minute. And we have our bill in the Senate, and we need unanimous consent. And we had one holdout. And so I just reached out to strategic folks, and they let it be known in their groups, like, we need this help. And I don't recommend doing this at all, but I'll share this. The staffers are overworked and underpaid. You do not want to inundate them with useless stuff, but we wanted this bill passed. And we had, I'm sure some of y'all are going to cringe when I share this.
Some of us may have done this. We may have done it in a different way, not publicly, but we'll say that.
We just had folks reach out and said, please co-sponsor. And I don't recommend that because I'm blackballed from that office. You don't want that to happen. But, you know, through that failure, because we didn't pass it, we've got so much strength within our community moving forward. And so I'm not certain when you want me to speak about what's going on, but I'll share what's come of that and what position we're in a little bit.
Yeah, I'm going to ask Ellen to just give an update on where her bill stands at the end here. So you touched on something that I wanted to hear from everyone about, which is when we have urgent communications that we want to send out, how do we decide how to communicate that to our community, and how do we vet that information to make sure that it's accurate?
That's a fantastic question. I think I tend to be more of the philosophy of you kind of get advocates really engaged when there's something that needs to happen pretty urgently. For example, last Congress, we were working on passing the Childhood Cancer Star Reauthorization Act, and we were in a very similar position as Ellen was. It was end of year, things were getting tough. It was like right about to be Christmas, very, very tough timing for the end of a Congress. And so like the last 2 weeks of December, we knew like it's now or never. And so that's then when we activated all of our advocates, and strategically within the Alliance for Childhood Cancer, we tried to share common language among our organizations so we could all speak about it in the same way with the same goal. So we're all asking for final passage. We're no longer asking for co-sponsorship. We just want this thing to move. Or like we were getting word that the House Energy and Commerce Committee had some final questions. And so who do we know in New Jersey? I don't know somebody, but this coalition partner does. Maybe they can get an advocate to call Congressman Pallone personally, and just trying to figure out who knows who and who knows what that's the most valuable kind of at that very moment to pull the right levers to make something go. But a big piece of that too is having that shared common language that we're using and working together on that. And so, things that were on like the St. Baldrick social media page matched what was going out in an email, which matched what some of our other alliance partners were using too, with the exact same verbiage, which also then means that the people on the Hill are hearing the exact same thing in the exact same way from all different facets of the community, which suddenly then resonates because it's consistent, they're hearing it over and over again, and it's not muddied in any way. We all had the same thing, pass star. And so that made it very clear from everybody what the clear goal was and how we could communicate it successfully.
Yeah, and I think an important point to make is if this feels like it's a lot of information and you really don't know where to access this information, all of us are doing work on this. And just come up to us, talk to us, ask us how you can become an advocate. And again, a lot of us have initiatives that are focused on maybe things that you care about. So just come up and talk to us. I'd love to hear from someone about how they deal with disappointment, because inevitably it's frustrating, especially with the way things are right now. How do you deal with that disappointment, and how do you make sure that you can bounce back and come back and continue to advocate?
I use the analogy of advocacy is much like research. There's a lot of failure, and sometimes you have to have a lot of failure until you finally succeed. But you have to put the time, the energy, the money, and other, as an organization when you're dealing with it as an organization. But you have to put the time and effort into it and fail, and be okay with failing, because there may be things on a piece of legislation like the original STAR Act. It didn't pass the first Congress. We were at that 11th hour, and it didn't pass that first Congress. And it was so incredibly frustrating. I mean, I will tell you, I was in tears in Florida, sitting on a dock going, I cannot believe it did not pass. But, we worked so hard to do this. But again, it's this persistence. And if nothing else in this room, we know that our community is persistent. And it shows. We've done, this community has been one of the most successful, disease communities in the last 10 years on Capitol Hill. It is often unheard of that a disease community passes specific legislation for itself, with our champions in Congress, almost every Congress for the last 5 sessions. That is unheard of, and that's because of people like you who have been involved with us, who work with the Alliance, who work with all these organizations, who work with individuals who are working on particular legislation. That's because of all of you, because you're just as persistent as everybody else.
I think I'll add too on the disappointment front. I think for me, one of the pieces I really value and how I kind of manage that is community. And so if I know that there are other people I've worked with and we're experiencing the same kind of disappointment together, it's, okay, let's go grab a cup of coffee and just kind of have our little wallowing session, and then the next day we're gonna wake up and try again. But knowing that there are other people that you can lean on. And then the other piece that I find helps me a lot is being very secure in knowing I did everything I could to get what I wanted as far as it could go. So knowing that I did it the right way, that I was clear, I was persistent, I had the right information, I brought in community partners, we all kind of did our best, and sometimes the forces are beyond us, and that's just kind of the nature of advocacy. And sometimes it all lines up and it's magic and wonderful things happen, but knowing that, we did it together and collaboratively, and really it was a team effort, and we all are sharing in the successes or the failures, it really helps me kind of process it too and kind of get up the next day and try again.
I think, too, it's really important to remember that advocacy is a marathon and not a sprint, because we're so passionate about our cause or our bill, and we really want it to pass, and we think that we've done great. I remember when we started first advocating for the Department of Defense, and to Ellen's point, I just want to say, I was just a friend whose child had a medulloblastoma, and I went to the hospital, and when I saw how sick William was, I could not believe the dichotomy between William and Jonathan. And I looked at William's mom and I said, you have to tell me what to do to help you, and I will not make you dinner. We are beyond dinner here. Now I call her sometimes and say we need to make dinner. That was 15 years ago. I didn't have any experience in running a nonprofit or in advocacy at all. So you can learn, I promise you. That is it, you know, Ellen learned, I learned. These two
I learned.
Yeah, I mean, really, so just know that. Nobody's up here with a big policy degree except for maybe Sarah.
No, I have a health policy
Okay,
degree,
there you go, there you go. But we don't. I have a journalism major, you know, here we go. But it is a marathon, not a sprint. When I first started with the DOD, I had to write a white paper. I had to ask someone, what's a white paper? Y'all, it's a research paper with footnotes, that's it. But you want to source every other one. You want to use your highest source, that's the NIH. Okay, so you got that. I wanted $30 million, just $30 million from a $1.5 billion budget. How hard is that for kids that are dying of cancer and for our servicemen and women who are ages, 15 to 39? First year in, we got $3 million. I was so defeated when I heard that. I mean, I was so sad. And my member who was helping me called me and said, oh my gosh, like this is so exciting. No, and I'm like, why are you excited? It's $3 million, we want 30. And he was like, because nobody gets money the first time in. And he said, let me remind you, this is a marathon. This is not a sprint. You have basically done a 5K. Like, we got a lot more work to do. So just remember that. It's these small, sometimes even your small victories really seem like huge disappointments, but they're not. And it is a marathon, not a sprint. So just remember that.
You know, it's probably a great time to plug the session that's going on right after this across the hall, which is about making sure that you're watching out for your emotional well-being in advocacy work and making sure that you don't burn yourself out. So that's gonna be right after this. I want to leave some time for those updates and then maybe some questions in the last 10 minutes. Ellen, can you tell us where your bill stands right now?
Yes.
And then maybe, Sarah, you could
sure
give us an
will.
update on kind of the broad view of everything?
I'll kind of start by talking about disappointment because talking about a marathon while you're sprinting. But we all know disappointment. Every one of us in this room. I mean, we're disappointed that there aren't better treatments for our kids. I mean, we could go on and on with the disappointments that have happened because of the diagnosis that our kid or sibling has. But I always say when I'm talking with members that my daughter died 50 years after Neil Armstrong's daughter died from DIPG. 50 years. And in 50 years, we'd gotten men to the moon, rovers on Mars, we'd had a car orbit our Earth, but we couldn't solve something a couple inches under our skin. I mean, talking about disappointment, right? Still the same prognosis. Phase 1 clinical trial and 6 to 9 months to live. But you know what? Now, with some of the new discoveries that are out there, the kids are living closer to 2 years. And is that good? Yes and no. Yes, because we're further along, right? But that's because of us advocating. And not just at Congress, but also to those researchers that are out there, right? And so, on that note, kind of segueing into what's going on with the kids first, I could have stopped. I could have just, you know what? We got $126 million. That's amazing. They're doing great work. But you know what? I made some amazing relationships with the people that received grants and with the folks at NIH and NCI. And, I get frequent calls. I think Adam is in here. From him, he's like, what can I do to help? And you know what? This is the last year. You need to get more money. Do you think that I could let Adam down? I mean, no. And so what happens is we develop these amazing relationships, and so they help us keep going. And so on that, with the relationships and what's going on with our legislation, as I said, last Congress, 11th hour, one person kept us from going over the finish line. We reintroduced in both the House and the Senate simultaneously, very bipartisan, which has been fantastic. Almost immediately in the House, it went to subcommittee and full committee and passed both of them unanimously. So we've moved on to the next step. Many bills, the majority of them don't get through committee. So that's huge for us. I was actually texting some folks earlier to find out today in the Senate, it is being marked up, and so very excited about that. And so then what happens from that point is we need both the House and the Senate to move on to a floor vote. And then once that happens, and both sides, the House and the Senate, agree with what the language is, then we move on to having the bill signed into law. And so I'll share a couple of things. When we started this Congress, we were asking for a reauthorization of $25 million a year for 5 years. In the House, they said that they would go with the original dollar amount, which is 12.6 for 5 years, and it passed. And so I'm thrilled with any dollar amount. So that's where it stands in the House. But in the Senate, they worked and they got approval from Bernie Sanders, which is good because he's the chairman of the Help Committee, that it will be for 10 years, starting at 12.6 and escalating up to 25 at the end of 10. So, put good mojo out into the world. That's right. So we need that to pass, and so pass the committee. And so once that happens, like I said, they each need to go to the floor for a vote, and then the House and Senate will come together. And so my ask of you all is come up to me. I'll help you in every possible way, but find out if your elected officials have co-sponsored, and if they haven't, I'll give you the one-pager that you could send on to them. I'll join meetings with you, share your story, get folks that are in your state or in your district to reach out with you and tell them the importance of needing their support. And you know, here's to, you know, lots more money for research.
And then amazing news, first of all, and I love that it's like up to the minute, which is always so exciting. And then in terms of the rest of the year and the kind of bigger picture, I think, I mean, I'm a little bit of a news junkie. Hopefully some of you watch a little bit too, but the big kind of elephant in the room is the federal fiscal year ends in 9 days, and there is no plan in place to keep the federal government funded. Even just this morning during the Childhood Cancer Caucus's annual summit, there was a vote on a defense funding bill that failed again despite some assurances it shouldn't. So it's a little bit of a mess right now in how that all moves forward. And I think while I hate saying things are inevitable with the government shutdown, I think it's feeling more and more inevitable as the days go on. And then you get into the like, how long does it last and what does that mean? And I think we all know for research and for NIH and the National Cancer Institute, a shutdown is terrible, especially for anybody receiving treatment at NIH. While a lot of those services continue, it's often pared down staff, it's limited resources, even things like cafeterias being closed. It's just miserable for everybody and completely avoidable, and I say that with my nonpartisan hat on. So that's the big piece. And then I think then the next piece is, how do we get out of a shutdown situation? How do we resolve the funding situation for the next fiscal year, which will be late, which is kind of typical, but still kind of frustrating in how the process works. This year compared to many years past, for the last decade plus, the kind of limits within which Congress is working to fund the government are much more limited. So the expectation is that there won't be as big of funding increases for the National Institutes of Health or the National Cancer Institute in the year ahead, which is really difficult. And this is one of those when we talk about, how do you handle the disappointments? It's not because we didn't do everything we could to push that forward or to say we still care about this. We went in with the exact same amount of passion and energy and facts and united information messaging as we've done every year, but there are forces beyond us, and this year the forces that are really pushing more fiscal restraint are winning a little bit this year, and so that's just something that I think we all have to get a little bit comfortable with, for better or for worse. But really, between now and end of the year, it's really resolving the federal funding piece is what comes next. And I know, Danielle, you have a brain tumor bill coming up too.
Right. So first, we have Head to the Hill on the 27th, and although registration is closed to actually participating in meetings, there will be an action alert that talks about appropriations and shutdown and making sure that the federal government understands how much it would impact our community, the brain tumor community as a whole. So I encourage you to look on the 27th for an action alert on social media. And again, on Ellen's bill, we'll be doing an action alert as well in the coming weeks. But we have been, for the last 2 years, working on a brain tumor bill, much like a STAR type bill. And we're been focusing on working with all of the stakeholders in our community to learn what's needed and to what kind of policy solutions exist that haven't passed. It's kind of going back to the basics of what we did for STAR, learning what ideas are out there, what ideas hadn't passed, what could potentially pass, and we've put it together. We're in the process of final tweaks, and we're hoping that in the next month or so that we'll be able to share some more information about a comprehensive brain tumor bill for our community.
Yeah, I think the bottom line here is we're heading into an end of year that's pretty uncertain. This Congress, I think, has been one of the more bewildering Congresses I think any of us have ever seen. So if you're worried about where to start or where you're getting your information or how to navigate that uncertainty, just come up and talk to us, understand how you can become an advocate, make sure that you're getting information from the right sources. I know and trust a lot of the organizations up here, and rest assured, if you come to us, we can keep you in the loop. I don't know that we have really any time for questions, but if you have questions, please come up to any of us and ask, and hopefully we can answer. I'll be over at our table over in the next room, and we really need your help towards the end of this year. Again, there's so much uncertain, but with your help, we can really make sure that our legislators and our partners are listening to us. So thank you so much for attending this, and I encourage you to go to that next session across the hall.
Absolutely.
Thank you.
Thank you. Oh, yeah, there was Pete's brain tumor, and there's larger cancer community opportunities, yes. It has
No.
see if you got it. Yeah. Oh, good, good, good. Okay. So just sit back. Oh, yeah. Oh, would love it. But even afterwards, I mean, that have my cell. Just text me.
everybody for joining us after the break. My name is Lauren Polovich. I'm the Associate Chief Medical Officer at the Focused Ultrasound Foundation. If you don't know about us, we do have a booth in the other room, so please come by and visit our table and learn more about what Focused Ultrasound is doing in the pediatric brain tumor population. I have the distinct pleasure of introducing our panel on advances in imaging and analysis today. This is a great expert panel, which is going to be kicked off by Dr. Nabavzada. Try to say that 10 times fast. Another, just an excellent panel here to talk about different advances in imaging that are quite new and maybe unfamiliar to some of us. So we'll start with about a 15-minute PowerPoint presentation and then move into the panel discussion. We have Dr. Davison, Dr. Ismail, Dr. Resnick, Dr. Koshman, and Dr. Mehta, who you guys have already heard from a little bit today. So please join me in welcoming them to the stage. Thank you so much, guys.
All right, great. Thank you very much for the introduction and the organizers for inviting us and giving us the opportunity to talk about imaging. So I'm going to present a brief presentation, 15 minutes or so, about some of the work that we have been doing in CBTN in the space of pediatric brain tumors, and then some of the challenges that we're trying to address, and then that would be a good segue to start the discussion with the panel members. And so going to focus on some of the tumor segmentation efforts, some of the efforts that we have been doing on machine learning and AI to better understand the tumor molecular processes and immune microenvironment, and some of the ongoing efforts and existing challenges. And so I start by showing a brain tumor, and as you can see, unfortunately, brain tumors in pediatric population, and in general, they're not like just round structures that are easy to measure. You can see that this tumor is in the thalamus and basal ganglia region and has like multiple different components in very irregular shape. And the current practice for radiologists is to just measure them in 2 dimensions, right? And you can see that we can have 3 radiologists measuring the same tumor very differently, here and here. And if you're using these measurements in the context of clinical care and clinical trial to assess treatment response to experimental agents or standard of care treatments, then that would intervariability to the process. And so you can see another example of a patient with medulloblastoma in posterior fossa, again, a tumor that has multiple components. And again, you can see that different radiologists can measure this tumor very differently compared to brain tumor segmentation that provides the volumetric measurements of the tumor, and not only the tumor as a whole, but different tumor components. And you can see that also there is some tumor in here in the internal auditory canal on the left side, and some leptomeningeal tumor that was detected by the model. And so brain tumor segmentation is very important in treatment planning, response assessment, in radiomic studies, which is actually using images as a quantitative metric, and also response assessment. And but it's challenging to develop a pediatric segmentation model because of lower incidence of tumors in pediatric, heterogeneity of the data, and also lack of multi-center data and data scarcity. And so one way is that can we use adult models in pediatric? And then the answer is probably not, and we tried it and it didn't work very well. The reason is that, as you can see, the brain in pediatric, in a 3-month-old, is very different from a 1-year-old and 5-year-old and 18-year-old and young at adulthood, because the brain is developing and the gray matter and white matter is developing. And so the models that have been developed in adult don't work in pediatric. And another major difference between pediatric and adult is that pediatric brain tumors frequently have cystic elements that are not captured in adult models. And so at CBTN, we developed a model based on multiparametric MR using different scanners in a variety of histologies and multi-center data. using deep learning, and this model has been developed, and the paper was published last year. So you can see that in the internal test data and external test data that we have MR scanners, the model was able to do accurate tumor segmentation. And the DICE scores for tumor as a whole is very high. And DICE score is the overlap between what the model segments and the ground truth segmentation by expert neuroradiology. So this model worked very well, but we consider this as a work in progress. Since the initial paper was published, we added certain features to the model, including adding the pituitary to the brain extraction mask, because pituitary is a common location in pediatric tumors and is not captured in usual skull stripping, and also added the defacing module to it to be able to de-identify patient's face. And this model is now available in our imaging platform, Flywheel, for CBTN researchers to be able to use it in the research. And so when we have volumetrics, we would be able to generate growth curves for tumor, which is very important because, as you can see, the tumors are frequently multifocal, irregular, and sometimes one component of the tumor starts to grow, like this component you can see here, starts to grow very slowly. However, for the interpreting radiologists, it might be very difficult to detect slow growth. It's like watching the grass grow. It's very slow, but if you look at it from like a few years time perspective, the growth is obvious. And this is actually a quote from the radiology report that the residual tumor is very irregular in shape, making precise reproducible measurement difficult, which is actually an accurate statement. And you can see that this patient has been imaged across, this is days, have been imaged multiple times, but the growth wasn't captured until the fourth time point, and the tumor was continuing to grow in this component of the tumor. But when we look at the volumetric measurements, we can see that the volumetric measurements can capture the tumor growth much earlier, and the tumor was growing along all these imaging time points, and it wasn't detected because it's just too difficult to measure that, especially if the radiologist is only comparing the imaging with the most recent one. And along the same line, we are working on developing an infrastructure for this imaging segmentation to be able to be utilized in the context of clinical trials. So sites could upload their imaging, and then we de-identify them and store them, and then route them to our imaging core lab, and then do tumor segmentations and volumetrics and send them back to the sites so they can have near real-time volumetric data for response assessment in clinical trials. And so this is an ongoing effort. This is going to be a multi-year process, and something that we're kind of looking for funding through various agencies to be able to achieve the goal. And so I'm going to focus on a few studies that we have been using machine learning to inform on molecular processes in low-grade gliomas and biological properties of these tumors. And so this is work that we have done last year and is now published. And basically, we use imaging features, basically the signal intensities that we can extract from the MR images to be able to do unsupervised clustering of pediatric low-grade gliomas that are on the surface have the same histology and tumor location. So they basically, we can put them in 3 different clusters. And so in this preliminary work, we have shown that the key tumor mutations have different frequency in these imaging clusters. But this was just the first step. And subsequently, we increased the cohort size and also collaborated with our bioinformatics colleagues, Adam Kraya, and the imaging lead for this work is Anahita Kazeruni, the data scientist in our lab. And we were able to actually see that the genomic profile of patients in different clusters are different. For example, if we compare the cluster 1 and 3, we can see that the regulatory pathways of pluripotency and differentiation was upregulated in cluster 1 versus cluster 3. And cluster 2 and 3, again, some important differences in their biological pathways. And we also did additional analysis with gene set net correlation analysis. And again, we saw significant differences between the clusters. So something that I want to briefly touch on, that by looking at the imaging, we can look into tumor biology and understand the biological processes. And if we basically test our model and show that it is accurate to predict these processes, potentially this can be used upfront in precision clinical trials, so the patients could be treated with the appropriate agent even before having biopsy or surgery in a neoadjuvant basis. And we also looked at prediction of tumor immune profile using radiomics. And basically this time we started with RNA-seq data, and we basically used unsupervised clustering, and then we used radiomic and feature extraction from MRI to see if we can predict the clusters of different immune clusters. And so you can see that the low-grade gliomas had 3 different immune clusters in terms of the immune score, macrophage, and microenvironment score. And the BRAF mutant low-grade gliomas exhibited greater immune cell infiltration, and despite not having significant differences in tumor mutation burden, the tumor inflammation signature was higher in cluster 1. And what's related to imaging is that imaging showed initial promise to predict different immune clusters. And the AUC is not very high here, but this is based on limited sample size, that we're increasing the sample size and using more advanced features, hoping to improve the accuracy of the model. But we just have to understand that if, for me as a radiologist looking at MR images, there is no way that we could predict like immune signature of tumors. So this is really promising data and a good start, and we're very excited about it. And finally, we looked imaging to be able to predict progression-free survival in patients with low-grade glioma. We know that pediatric low-grade glioma is a heterogeneous disease, and based on the clinical and genomic data, we can put the patients on low risk, medium risk, and high risk group, which basically imply different treatments in them. However, we were able to combine clinical data and radiomics to be able to kind of do the same thing without having basically histology, and come up with a stratification of patients into high risk, medium risk, and low risk based on the clinical radiomic data for prediction of progression-free survival. And so now I'm going to focus on some of the basically challenges that are existent in the space of data. Obviously, there are data access issues and also technological and computational barriers in data processing and heterogeneity of data, and all of these would lead to lack of reproducible models. And so we need to basically streamline the whole process in terms of transferring medical imaging, image preparation, and pre-processing, and to integrate the imaging data with other data in terms of genomics, proteomics, and clinical, and finally harmonize it and use it for scientific discovery and clinical translation. And a couple of things that we have achieved in the past couple of years is that we developed this segmentation brain tumor model, and now we can use it in our imaging platform, which is a flywheel, and we can run it, and every CBTN researcher can have access to this tool for tumor segmentation. And we also tested an experiment throughout the Include initiative. So currently NIH Cloud Workspace is maturing. However, basically there is a lack of communication between different sectors. For example, we have Cavatica, which is our genomic platform. However, there wasn't any communication with the imaging platform, which is Flywheel. So we tested an initial cohort to be able to basically create a cohort in Cavatica, the imaging platform, and import it to Flywheel. And then so Flywheel could curate the cohort in terms of their imaging and also do the pre-processing and send it back to Cavatica. So the investigators that are using genomic data would be able to also have access to imaging data and do a more comprehensive study. This is another effort that we had last year, and we were able to bring digital pathology into Flywheel in the DICOM format, and also to be able to develop some gears or algorithms to do pre-processing for digital pathology. And this is our recent review about this process and different techniques. And finally, I'm going to touch on very briefly on the ongoing clinical trials that we have at CHOP using fluocyclovin PET, which is an amino acid PET tracer, and we're using it for response assessment in low-grade glioma, and also to differentiate progression-free survival and tumor progression in high-grade glioma. This is the first time that this tracer is being used in pediatric, and we recruited our first patient, and it was a success and no side effects. So this is something that we're very excited about. And this is the first patient, as I mentioned, that you can see very nice activity in the tumor, almost no brain background. So this is a very powerful imaging technique that have very high signal-to-noise ratio, and hopefully could help patients to have more powerful imaging compared to the current MR techniques. And with that, I would like to thank all the lab members and CHOP and CBTN investigators who have been helpful throughout all these initiatives. Thank you. All right, great. So I think we're gonna move to the panel discussion, and you can take questions from audience too.
Pseudo-progression. What's the machine learning pseudo-progression?
Sorry, the question is that how can we identify that?
What's your experience with pseudo-progression using AI?
So pseudo-progression, in the context of high-grade glioma, I think that's the question, or any specific tumor?
Imaging changes during treatment, differentiating treatment versus progression, pseudo-progression.
Sure. So there are different ways to do it. We can use PET techniques, molecular imaging techniques, with this is something that the clinical trial that I mentioned, this is something that we are actively doing. In terms of MR metrics, there are different techniques to do it. One is using physiologic MR, like perfusion MR, which is something that we have access to and is better than conventional MR, but the accuracy is suboptimal. The other method that we can use is using AI machine learning approaches to.
Have you made any progress with AI for differentiating?
I would say that significant progress have been made in adult, but not yet in pediatric, because we just don't have large enough cohort in pediatric yet to train those models.
Have you published any of that?
So I don't think there is much in terms of larger studies in pediatric, but certainly there is in adult glioma. Sure.
I wonder, Ali, if you spoke about models. I wonder if, just to make sure that the audience is on the same page, when you say that there aren't large enough studies yet to train models, what does that actually mean? Like, what's the process of creating a model?
So the process of creating a model is that we need to have a large set of data to be able to do training on a set of features that we extract from MR sequences. And so we want to make sure that whatever we train, it's reproducible and we're not overfitting the model, so to speak, because, and so we have to train the model and then test the model. And ideally, we should have also external cohorts that we further test the model on. So it's a process that requires large data set, and we also have to take into account for the heterogeneity of the data. And it is something that I'm hoping that we can talk more throughout the panel discussion, as some of our speakers really have focused on this area. So more to come. Right. Any more questions from the audience? Yes, please.
John Ligon from the University of Florida. So I appreciate what you're saying about needing larger groups of patients. How large are we talking about? Like, how many patients do you need to be able to accrue onto a protocol in order to be able to build these kinds of machine learning models? And I think in a similar vein, based on what you guys are seeing in adults, can you extrapolate across different modalities of treatment? Like, can you use data that you learned from a chemotherapy trial for a response assessment and extrapolate that to an immunotherapy clinical trial and extrapolate that to targeted therapy? Or do you need different data sets across different treatment modalities?
I mean, that's a great question. So how many data do we need? So I think we need probably a few hundred to be able to train a proper model. We can do some transfer learning in some areas, but not in everything. And I think that depending on nature of different treatment agents, I think that probably not all of them are kind of not translatable to each other. So I think that we just have to be very specific. One thing about pediatric is that from a biological perspective, it's very diverse, so it's a very interesting field to study. Unfortunately, that also makes it for radiomic studies and AI, ML more difficult compared to adult, that we have mainly glioblastoma as the most common tumor, so we have access to much larger data sets compared to pediatric, that the histologies are much more diverse. And so consequently, the number of patients, and also because of the lower incidence, the number of patients are much smaller, which really opens the door to the need for multi-site studies and really everyone working together, using all the tools to bring every single data point together to be able to train these models. All right, great. So I think we can ask questions from the panel. And my first question is for Dr. Resnick. And with regard to the precision, basically tumor boards that is currently ongoing, and basically the notion is that how can we use the data that we already have from the patients to predict which type of experimental treatment could be used. So how do you see that in the context of adding imaging, and both in terms of, because imaging, we don't necessarily have to wait for the biopsy or surgery to happen. We already have it after we have done MR. And also in combination with the basically tissue-based data, how do you see that process kind of working and also changing the landscape of treatment?
Yeah, were you in the audience when Elaine Martis was presenting, by any chance?
I was.
Oh, you must have missed it.
Yeah.
So you missed the point where Elaine essentially said that she thinks radiologists are going to be out of a job pretty soon. But she was being partially facetious in ways that I think ties really well to what you presented, which was that she was highlighting that taking liquid biopsy samples, collecting blood samples from patients who have been diagnosed with a tumor, they could start detecting changes in circulating DNA in the blood before the radiologist could see changes in the MRI, which I think is very validated by what you just presented, right? Which is that, the human eye essentially can miss changes in the images in ways that we can augment using these technologies. So I'm not an imaging scientist. I really focus, mostly on the molecular changes that underpin the biology of tumors. But for me, the really exciting part is a recognition that within images is information that is not interpretable by the human eye, but that the machines can essentially leverage and be trained on those images to augment our understanding. So Elaine made a really great point to say that, when you start adding different layers of data, you can cross-connect across those bits of information from methylation to genomics to imaging in ways that can support our understanding. The key part about imaging as related to, I think, the molecular tumor boards framework is that in contrast to the single point in time that we get in the sample for a biopsy, typically, images, as most of you know, are taken constantly, right? So you're essentially constantly collecting data in a longitudinal way that is a perfect space to augment the way we think and understand the way tumors change if it's brought into the analytic space. And I think that's the big gap of opportunity. When a radiologist is looking at images, they're not sitting in a computational environment. They're sitting right in their office, looking at the film. They don't have a computer who's analyzing and doing AI in it. And I think that's the opportunity to create these real-time analytics that can drive this augmented analysis. To the point where, and I think we can envision a future where, it's not just clinicians who are uploading data, but, you can imagine you yourself, right? As patients who you have access oftentimes to your imaging data, you can imagine yourself uploading those MRIs into a platform. And for the AI to just provide a framework to say something, right, that you can further interpret yourself even as a family. Now that's, in a future state, but those are the kind of places that we can see sort of augmenting the decision framework for both families and for clinicians, radiologists, in ways that can intersect around a decision-making framework. I mean, I was just sitting yesterday at a conference, and the IPG family came up to me, and they said, what I'm sure Carl and everybody sort of hears all the time, right? The next scan is X, right? And, we're terrified, by what the next scan is going to show. And so the only way to combat that is with more information, not less information, right? So you have to, this is the hypothesis, right? To maximize the amount of information we can extract from every bit of data that's generated in ways that can inform ostensibly both the family and the clinician about what's taking place. So that's the hope, I think, that these types of technologies present. The last part I'll say is that, I think you mentioned the difference between the adult landscape and the pediatric landscape. And I think you're right that, the opportunity space on the pediatric side is, from an AI perspective, is so much larger, right? The diversity of data and the complexity is the perfect space for AI in ways that is not as prevalent on the adult side.
Perfect. Thank you.
I'll just give Carl a chance to mention, because you actually treat patients on the tumor boards.
I don't think I can improve upon your speech on the topic. I will say that, I've sat with a lot of families, even in this room, really struggle with this pseudoprogression question. I think probably the key central radiologic problem with DIPG and DMG is this. The tumor looks a little different. I can't tell you if the tumor is growing or if that's radiation effect or some combination of the above. And I think one of the things that we struggle with is, I've been going to conferences for a decade where I've seen really exciting things. And this concept of 3D is such a natural concept. When we have trainees come into the clinic, they say, why are you doing it in 2 dimensions? And it's like, well, we've been doing that. That's just what we do. And it's such a natural fix to go to 3 dimensions and have a better sense of how the tumor's changing in every dimension. It's not a sphere, right? It has these odd shape, and it needs to be monitored. I think there seems to be a particular lack in radiology of moving the novel innovations into the clinic. There's like we are stuck doing two-dimension MR, and that's where we've been for 2 decades. So I really like seeing these talks, but then I'm like, what is it going to take to get it into the clinic? And one of the ways this works in genomics is if another center or a send-out or a liquid biopsy platform has a better option, then you as the patient can then just have your tissue sent there. The field moves because of that natural selection. If you have an MRI and your local team gives you a two-dimensional read and can't tell you whether it's progression or pseudoprogression, there's no market available right now for being able to say, well, I just send it there. I think the things that Adam and Ali are talking about will get us closer, right? So if you as the patient or you as the provider can tap into a system and say, I'll just submit the files and you give me the 3D, or you give me the AI on whether this is progression or pseudoprogression. It seems like we're right on the cusp of it, but I'm also like jaded by how slow it's been and how I've heard really cool things 5 years ago and they are not in the clinic. So I'm not that person to make that leap, but I think you guys are closer to being those people, and we need to somehow get the logistics, let the rubber hit the road.
Great. Yeah, so actually, anyone else on the panel want to? Go ahead.
Talk about needing more data for models, and we talk about where that could go. So many times as a parent, you're like, what can I do? What can I do to help you? Because I am not smart like you. And I just wondered if, we just opened up the DIPG One link last week, a platform where they can submit all of their data, including images, to X-Inform, as well as connect with CBTN. Is there a format in which you would want to make sure those at X-Inform are receiving it so that you could use it, because it is openly available to academic and government ecosystems without cost?
Yeah, absolutely. I think that's really a great step to add the data that we really need. And so I would say that the more comprehensive the MR is, in terms of having the right imaging sequences, the basic imaging sequences that we base the model training on, the better. I mean, thing is that, and I think it was kind of mentioned that by Carl in some ways also, that a lot of times patients don't appreciate that the added value of better imaging. They view MR as like a commodity that, okay, I'm going to get MR. It's like buying a Big Mac in McDonald's. It's the same thing, right? But it's not. So it's really, if you have good imaging in terms of volumetric imaging at 3 Tesla MR, that is much more powerful compared to an MR that is done on lower magnet with like thick section. So I would say that there are two elements, really having high quality imaging and having the complete set of images. Those are the things that would, down the line, help us to develop higher quality models.
And as I'm listening to this, I'm wondering, Lisa, that, as I'm listening to Ali give the answer, like, even if you ask me, like, is this good imaging or bad imaging, right? It may not be obvious, right? And so I wonder if that's something we can do better at, to communicate to the community and say, like, even if you don't know what any of these words mean, right? If you ask, is my imaging this, right? Then that's an opportunity to ensure that the data being generated, right, meets that multiplicity of use. And, if you have a choice, essentially, as a family.
That's exactly where I was going. So just as a follow-up, 2 years ago at the Tough Together breakfast, Dr. Mueller unveiled, by consensus of many of you, the new best practices in DIPG. Is it possible that over the next few months you could develop together a consensus on what is the imaging we could come to expect, and then we can roll it out through my DIPG Navigator and on the DIPG One link. So patients who are hit with this diagnosis can absolutely know this is what I should be asking for. And I hope it's not just for DIPG. I hope that's just a model for every other tumor type. But we don't know what to ask. And 2 years ago, we were, starting with 4 milligrams of Dex and didn't know any better, right, until those best practices came out. So thank you so much.
Absolutely. I think that we definitely can develop these guidelines as a part of like the imaging core. And so definitely it's something that we're going to provide.
Can we follow up on this question? There's a
Sure.
challenge for patients to be able to know what type of files to actually download and how to download them. Sorry. So it's not an easy thing for patients to be able to get their DICOMs and send them to a site. So if there's a portal that supports the family to know what images they're interested in and in what format to get them in. I mean, I trained at CHOP as well, so in places like this, of course you expect the best imaging and you just take this as a matter of fact. But if you go out into the communities, you have different type of imaging facilities. And I've seen patients that come in with JPEG files and individual like screenshots on their phone, things like that. I think if we're developing standards to do this, it would be great to have one place that has an overall protocol on how to go about requesting what files and how then to upload them. That would be hugely valuable to not just patients, but also to their treating neurosurgeons and oncologists.
Thank you. I can't agree more.
Yeah. And I think we envision, my uncle, maybe this was 2 months ago, actually passed away after going into a coma for reasons that weren't clear. And this was in a rural place in Maryland. And so I was trying to get the images, right, from the hospital just to get a second opinion. And the only solution that they had was they were willing, the nurse and everybody was willing. So they got on a Zoom call with me, turned the screen towards their own screen, and through Zoom, I was looking at their screen, right, at their MRI. And that was the only way, right, that they could figure out how to share the images, because otherwise they would have to put in a request to burn a CD to send it to us. It would have taken way too long. So this, we've highlighted, this is a huge gap in the healthcare framework, including to NCI. You heard Elaine earlier today talk about the fact that she can load her genomic data, right, in like 90 seconds, right, into the NCI. And imaging has, as a data format and infrastructure, has not been invested into the same extent by the research infrastructure as omics data. And so there's this gap, a difference, even though imaging is done as a standard of care and genomics is not. This capacity to leverage imaging data is still a gap. Now, there are technologies that make that possible, and I think that's a key movement, because it shouldn't actually be, right, the responsibility of the patients to try and get that image and figure out if it's a JPEG or a DICOM, and like that. You guys are worrying about many other things, right? It really ought to be part of that infrastructure, and it's coming along, but as I experienced, and I'm sure many of you have, there's a huge gap. But that's the way it really should be, is to bring imaging data to parity with genomic data in terms of the standardization around which data can flow and be utilized. It's digital, right? The fact that hospital systems still offer to burn a CD, that itself is an anachronism, right? Like, I don't have a CD-ROM, right, to even look at that, right? But it shows this gap, right, that between healthcare systems and their investment in data-driven infrastructure in comparison to almost any other data domain in society, right? Hospital systems have not yet fully recognized that they are data companies, not just healthcare companies. And so that's a really key opportunity. And again, something that Carl mentioned, right, needs to be driven by that selection process of the community beginning to push for what is acceptable and what's not.
So I'd like also to add something about this. So I'm a researcher, so I work a lot with computer analysis of images. So what happens is, so I'm an employee at a public university. So what happens is we start collaborations or data agreements with hospitals. Like, for example, Cleveland Clinic, when I was a researcher back at Case Western Reserve. So those data user agreements, they take months for us to get public access to the data or to get the data transferred over PACS to us to be able to analyze those images. But what we always work with would be routine imaging, like MRI scans, the different sequences, and the consensus in medical imaging is usually the DICOM format, and then we're able to convert this to whatever format that we can work with using our computer programs. But also there is, right now, avoiding those like the routine and getting these scans in forever time, and we need to move faster, obviously. There is now like a huge pool of public data that we can use. But unfortunately, this is not still moving forward with pediatric brain tumors in general, because it's a rare disease. But I think with in the future, this will change because of the efforts with the large brain consortiums right now, like CBTN and the other brain consortiums. There is some hope that we can work with public data sets and have like radiomic models or models that can be validated on a large group of patients and with just the available routine imaging. Like there's no fancy models here, it's just the routine MRI scans that every patient is undergoing when they're diagnosed with a disease.
Great. So I think this was a very great opening to a question that I'm going to ask from Ashwini, and that is, when we have a scarcity of data and also the timeliness of data transfer, I think that really opens the door to like some cloud solutions. If you can elaborate on your efforts on AWS.
Great. Hello, everyone. Ashwini Davison. Before I get into my current role with AWS, I will go back to your slide about the inter-rater, reliability and the challenges there, and mention that, yeah, once upon a time I was that house staff, right, at Johns Hopkins, and I'm the person, if I was in the reading room, that you would not want to be banking on when I'm 38 hours into being awake and, reading an image. So those studies with the inter-rater reliability were probably done with, apples to apples, let's say, faculty, but so many other variables, and that's where I do think that the power of AI in aiding that cognitive computing is going to be super important, Ali, in helping here. So Lisa, one thing that I would say is, especially with the amount of data that you're referring to, and all of the work that's going on with foundational models and a lot of excitement in medical imaging. Something that I think Amazon could support with is, Adam, and I was just mentioning it to Adam a little bit earlier, we just announced the new Amazon Research Awards applications. They just opened today. They're due the first week of November. So especially with this audience here, where there is, academia and passion, I would encourage everyone for the AI grouping, Amazon Research Awards. There's a cash award and there are promotional credits, especially for something where there's applications of ML for responsible AI or generative AI. Would be happy to support some submissions. They're due the first week of November. Today's the first day that they're open, but I think a group like this, especially if there is someone who has an important idea, there's this data, let's move quickly, and I think this community can certainly do that. So I wanted to mention that piece because the ability to, it's one thing if the family's uploading that image, but we're still in a world, right, where when that's uploaded, there needs to be the mechanism for another human being to view it, and how does that work, and how does that human being get paid? And, this isn't leaving out that radiologist, but in this particular use case, I think that there's a role that AI can play. So let's think big about that, and happy to support any submissions. on the AWS side, in medical imaging, one of our key areas of focus, a lot of what we're talking about, right, is this infrastructure for sending the image from here and then downloading it from there and then sending it back and making all of that possible, which is where things are. And, you know, the data sharing is important, but getting to that single copy of truth is also one of the real goals, right? Because the amount of data that's being duplicated, the amount of storage, the loss of fidelity, and as soon as you de-identify it, you no longer have that ability to update it and have that longitudinal view. So that's been a huge area of focus for us, Ali, in getting to that single authoritative, you know, source of truth and bridging that divide between the clinical and the research chasm so that, you know, for multiple use cases, you can point to that same image. So those are some of them.
Thank you so
areas.
much.
Oh, yeah.
Yeah, so sounds like you are from AWS. I was gonna ask that, have you looked at AWS Health Lake imaging? Because that's one of the key areas, and then they also have tools for natural language processing.
Yes, this is actually.
Right,
Yeah.
so, yeah, and what I was just describing there with the single authoritative source of truth, that is one of the newer services that we'd love to have folks pilot on and see if it's helpful here right now. The exciting, you know, update from, I think, a couple of weeks ago, right, Adam and Ali, we had submitted to support some supplements, and the teams were, I think, awarded the NIH funding to do some additional work with federated learning with imaging, and that's the flywheel work on AWS. So, and that needs to be done in a year. So hopefully we'll have some good updates by Brainstorm Summit next year on that.
So
maybe just to comment a couple of things just to make sure that, you know, we're using the same language. You know, what Ash just mentioned is really what I highlighted, that, you know, to do this type of analysis, like the 3D volumetrics that Carl mentioned, that you have to compute, right? You have to analyze that data. And the radiologist doesn't have a computational environment within their electronic health record framework. So you have to have a cloud-based computer, right, next door, right? Even Elaine Martis mentioned, right, that she leverages AWS, right, to scale computation for genomics. There's no reason why we can't do that for imaging. But I want to just pick on a thread that you mentioned, because you may or may not have been introduced to this term federated learning. You know, so one approach, right, is to bring images into a central repository, which is an approach that, you know, we've, you know, been championing in CBTN, and what you're hearing Ali and others talk about is to bring all these images into one place so you can analyze them and work on them. Another approach is to leave the images where they are, but send a computation to the images, right? So essentially, the cloud environment that Ash just mentioned doesn't require the movement of images. You can either move tools or move data, right? And I think then we can optimize on time. What takes less time, right? Can we just deploy the tools, right? And then the hospital doesn't have to move data, right? They can just do the computation and analysis there, essentially. They don't have to de-identify the images even, right? They can stay behind their own firewall. So that federated architecture is another layer of opportunity. And again, I think this is where the pediatric landscape is especially poised to transform the landscape, because, you know, we already need to collaborate. So establishing a federated architecture that's trusted by across institutions, where you send the compute, right, to those institutions so they don't have to move the images. You just get the results back, is the next layer that Ash just mentioned.
just one more question on that. So I'm a software engineer. I lost my son to DIPG. We have a ton of, I think, 14 MRIs over the period. I used the AWS tool just to play with the data. But now, where we are as parents, what can other parents do to help you guys get the data from the children who have passed? Is there any effort in that direction where we can go and upload these images perhaps to AWS, and then it comes to CBTN or through Flywheel or something else? Have you considered that scenario?
Yeah. It's a major initiative for CBTN, but let others all chime in. So when we first started within the CBTN, we prioritized biosamples because there was nothing known about the molecular components. And we initially only began collecting images for the diagnosis and then post-operative. But the reality is, for the kind of things that Ali just mentioned, you want every single image that's ever taken. So for the past 2 years, that's been a major initiative to go to each site and do this in bulk. So rather than, and this is what we're doing, essentially, to say, I'm going to go to Lurie and say, I want every single image for every single patient that's ever enrolled to come up. And it's been an undertaking at each of these institutions, right? The institutions don't have staff dedicated to just uploading data, right, necessarily. But bit by bit, right, those institutions, are supporting that process to the tune that over the last year, I think there's been more than 25,000 imaging series that have been uploaded. That's only still a fraction, right? We are still, so from our sort of organizational framework as a consortium, our job is to really maximize the opportunities that we can drive institutions to implement. Now that can pair, right, with a patient-centered sort of complementary approach where someone like you said, I have 13 images, right, on mine, how can I plug into that same infrastructure? And I think that's one of the opportunities that I think we're really excited to explore, and this is something Lisa and I talked about. Like DIPG OneLink is an example of that where you really harness the different workflows from maybe a patient-driven upload and institutional sort of framework and maximize and ensure that you can link across those efforts. So I think we're just at that very early phase of saying, how can we put all of these pieces together in ways that supports that uniformity across what is a patient can do and what an institution is required to do.
Sure. So Adam, this is Mika here from XYears. So on the OneLink platform, right, through XInform, I think patients have a right to get their images allocated, and we're happy to retrieve them. Yeah. So I think we've retrieved something on the order of imaging from over 520 plus patients. So these are, we basically go and get the images on your behalf from the institutions that they were at, fully identified in time series, and then if any researcher wants access to those, they can be de-identified. That takes a little bit longer. I think we've only de-identified probably around 200 or so. These are not the slices where there's thousands and thousands, but the actual time point series themselves. And I think through this initiative, right, we should be able to provide that data and make it basically accessible through Flywheel, or we operate on AWS, right? Through those networks, we should be able to make that data generally available for computation and modeling and generation. We've done work in AI, not around imaging, but others. But I think this is something that people are really empowered to do now, and it's just a question of find a way to do it. You don't have to do the work, you just need to know where to go.
I think it's a great opportunity, right? And you have essentially, someone like XYears essentially act on your behalf to get the data, right, at a one-by-one sort of approach for each of the patients. And that can complement things like the CBTN that's saying we want every one of Lurie's images to come in, right? And for those things to then come together as driven by a patient-centered approach, not just for DIPG and DMG, but that should be something we should do across brain tumors.
Go ahead. Yeah, I was going to say I'd love to hear the faculty perspective on something like that, and I'm eager to support and dive deeper into that, the imaging particular focus, because now that different infrastructures are there for a, patient retrospective approach, perspective approach, and the institutional approach, I think part of what this community can do is operationalize it and then make awareness and promote it to the faculty. Who are, doing a lot of this work to help raise awareness for what is available. So.
Yeah, I only do have one question though. Yes. So if you have patients collected from different places and they have different treatment protocols, like they've been treated, with different regimens, different radiation therapy strategies, then it's hard to analyze those images because it's going to be confounded by the different approaches and treatment. So I think ideally when you try to train a model or have your model learn from images, they should all be under a clinical trial maybe, like all the patients have been enrolled under one trial, they've been treated uniformly. And then when you get those images from everywhere, from patients, from, from public, from any public institution, now your model is robust because it has been trained on, uniform patients, and then it now can validate this approach on those external studies that have been collected from different institutions. That's from an analytical standpoint.
Yeah. So I think again, I'm not an imaging scientist, but I 100% agree, right, that essentially clinical trials provide a much more structured framework for that. The reality is, if you look across all tumor types, not all patients are on trials while they're getting their images taken. And so for us, the goal is to leverage all of that data, right? So, for example, we work closely with PNOC, a clinical trial consortium Ali mentioned, Carl's participating in. And on that side, we bring in the clinical trial data. But clinical trials typically are small, right? Number of patients that are on trial. So we have to also figure out ways to leverage the real world data. And this is where I think our hope is that to address the confounding factors, you have to have the clinical data, the treatment information alongside of it, so we can parse the data, understand what might be differences imparted by that in ways that we can intersect across clinical trial derived data, real world data. The other part that I would say, maybe Carl, you can comment on this, is that the pediatric, this is my perception, but the pediatric oncology community is actually fairly aligned in their practices in ways that the divergence in treatment is not astronomical. But Carl or others, please chime in on that.
Ali's telling me not to.
Yeah, I think that we have to close the session, but certainly there is a, are we out of
Carl
time
agrees.
completely? Yeah, so very interesting conversation, but to be continued, and I invite everyone in the audience to approach the panel members for further discussion. Thank you so much, everyone, for attending, and the audience for engaging and asking very good questions. Thank you.
And then, doesn't Carl stay?
Yeah, he's right there.
Okay.
Carl.
So you, Carl, who else are we missing? Just us? Isn't there one more?
Good to meet you in person.
Melissa, Mark. Carl, by Ann, Melissa, you. I guess there's room I can stay. Okay. Here we go, guys. All right, everybody. It is so good to see you guys having so many questions and conversation about where do we go to next? How do we put this in motion? This is how we came to this point today. We realized that our individual experiences were not all that individual. We just didn't have that community to bounce ideas off of and find resources together. And so if you were like me, you made inquiries on Facebook, or people reached out to you and said, I need you to know this or that or whatever. And at some point in time, as the people on this panel know, a 20-year-old in a hospital room in Stanford said, you've got to be kidding me. I can't even write my research papers using Facebook, and that's how we're making these decisions? I don't even trust you to do it, Mom. And so he called a Zoom call that Carl, I think, was on, if I remember right. I know Sabine was. And he said, my mom can't take these calls anymore because she's going to take care of me, and some little kid is going to get the ringtone. And I was like, yeah, that's true, because I am going to take care of you. And so with about 14 doctors on the phone, with Chad Tuff, Defeat DIPG, Mark Mosier on the phone, with Count Me In, with a number of folks on the phone, we had a discussion about how do you get to the best doctors when you're in Kansas, like we were. How do you get there? How do you know who to go to, who to trust? Surely there's something a little bit better than Facebook. And as we talked about it, I don't know that the doctors at the time knew, like, how did the patients get to them? You know, maybe there were feeder hospitals in their area, but maybe it was because they were opening a trial and they were the PI. But there wasn't really like a navigator focused on DIPG and DMG. There were some general ones focused on pediatric cancer maybe, or brain tumors, so you can maybe try one of those, but there wasn't somebody that dealt with it every day, who was learning how trials were going as they were coming out, who were seeing more and more MRIs, who were seeing more and more side effects to trials, and could do some comparison with kids. And so Mark Mosier, I'll never forget, called, even I think before Ann was hired, or right about the time.
Yeah, it was during merger.
Okay, during merger. And he said, you take care of Jace, and I'll figure this out. We need to do it. Tammy and Jenny, the two founding women, and Jason and Mark said, we need to do this. And they built a beautiful system we now know as My DIPG Navigator. And along the way, I remember Mark called and said, do we need to hire a nurse for this? And I said, not just a nurse, you need to hire Leslie Jared, our navigator. And luckily, he liked that advice. We then knew we had to do something a little bit more. And we have the best doctors, right? And they're all across the country, and they come out of PNOC and PBTC. They don't always know what's going on in every hospital because they're so focused on their own work, and they're amazing. But what would it feel like if you didn't have to call hospital after hospital for a second, third, fourth opinion? Because that pressure is on you, isn't it, as a parent of someone with a brain tumor? That pressure is like, is there something I don't know? Surely this cannot be what it feels like. Surely there has to be an answer. Surely this cannot be terminal. And that's a lot of pressure. It's a lot of sleepless nights, isn't it? And so, again, this kid was like, what if we just tried to figure out how they could have a Zoom call like I did when my ONC 201 wasn't working? Because they were really awesome to tell me I could do CAR-T or I could do this, and we didn't have to go all over the country. And so we started discussing a national brain tumor board. And Carl was one of the first ones that said, I like that idea. And Sabine and Jason Fangisaro and Nick Vitanza. And pretty soon it grew to about 12 of the best doctors across the country, with others saying, if there's ever a spot, please consider me. And yet we knew we weren't capturing the whole story, because we were putting all that data together to get it to the tumor board. But then what happens? Then you go on a trial, you move to another trial, you go home, maybe on hospice. We hope treatment's done and that tumor's nowhere to be found, and yet we know in DAPG that's rare. So how do we make sure all the data gets collected and used correctly? Because, if we think it's coming from, say, CBTN, maybe they're not able to collect outside their partner hospitals. So when we're taking care of Jace outside of one of the partners, that data isn't coming in yet. So we got back in touch with our friends at XCures who worked alongside ONC 201 back in the day, and we said, we got to do this. We got to collect this longitudinal data. We got to do patient assessments to know what quality of life there is. We're going to have to work with FDA to show that we should approve a drug that extends quality of life, even if it doesn't extend outside survival. Because if we get to pick, we want quality of life as long as we possibly can. We want to be out of the hospital, and we want kids on basketball courts and birthday parties and playgrounds. So we can't get there until we have the right data in place. And so I didn't put any slides up here for a reason. I want you to go to your back of your program. I honestly want you to do this. I'm not asking a ton. I want you to go to your phone. I want you to put this in Google, dipg1link.org. Okay? I want everybody to do it. I want you to hit bookmark, because what we just unveiled last week, it was a labor of love with a lot of the sponsors you see today, with Sarva, who lost her son, Mitchell, with Neve, who lost her son, Misha, who lost her son, Neve, with Stacy, who lost her son, Hudson, and me. And we tried to write, with the help of Salon and a number of people, Amazon Web Services, etc., helping fund, from new diagnosis to grief, what we wish we would have known. All of the resources, My DIPG Navigator, the Tumor Board, Gift from a Child, X-Inform, CBTN, where to find emotional support for your siblings, and a private community that's not collecting your data that we will build out over the next year, where you will find people interested in trials, a book club, moms or dads in the fight that just need some support. You'll find activities for kids. You'll find financial resources. And then we were like, financial resources. We got to do this better. And so Live Brave Together was born. We'd already been working together. But now we're, after 3 weeks, we're at about 25 to 30 foundations that will ask you to fill out one application online. You'll sign it online. If we need a social worker or a doctor's e-signature, we'll get it. And we'll fill a request as much as possible, whether it's requests for rent, travel to a trial, special experience, an earn, or some income support while you get back on your feet. And we're just getting that started. Lori Evans, many of you know from Noah Brave, has said, yeah, I'll take this one. And so it'll run through Noah Brave, but it will be a collection and a growing collection of foundations that have worked together since our sons were in the fight, honestly, that will do what we wish we had at the time. So everything you see that's come out of the last 2 years just came out of parents' experience like mine and like yours. And so if there's a piece you don't see, now's the time to say, but what about this? Because if you think we need it, we ought to have it, and we need you to bring it forward. Now, I'm going to turn all the rest of the time over to these guys to talk about what's happened after year one, and I want you guys to be thinking about questions you have as to where we go from here.
Amazing to see an incredible vision come to life the way that it has over the last 2 years. Hopefully my slides will show up here. So my name is Ann Friedholm. I have the honor of leading the Chad Tough Defeat DIPG Foundation, and happy to have the chance to talk about my DIPG Navigator. In case you are not completely aware what this program is, this is free one-on-one support for a family in the DIPG or DMG diagnosis and going through that journey, where we connect them with an experienced pediatric oncology nurse who's there to help educate, help connect to the key resources, especially medical, and to support all along the way. And all you need to do, in case there's a family in the room or you know of a family that could use it, is go to mydipgnavigator.org, fill out a very short registration form, and we promise that you will hear from us within 12 hours, but I can tell you our average response rate is less than 3. All right, let's see if I can figure this one out. I stand before you really on behalf of all of these beautiful faces. These are the families of Chad Tough, and I know a lot of our families are in this room today. So I'm really here representing them, our founders, the Carrs and the Mosiers, and our 38 family partners that are across the U.S. and Mexico. I can tell you all of us are very, very proud of the program that's been developed, and we find it to be just such a blessing to be able to have an answer when a new family comes forward and needs help, and we have an answer for that. I'm holding to make sure Stacy gets her photo. I also need to put a spotlight on our Navigator partners. We could not do this program without this group of partners. As you may or may not know, the primary focus of Chad Tough is to fund research. And if anybody's tried to say no to this lady and succeeded, please let me know how you do that. So she came forward, and all of our families, all those beautiful faces that you saw up there on that screen, they knew that this was needed. And so it is a priority for the foundation, but these partners here help us fund this so it can be free, while also funding as much research as possible. So, and I know there's partners in this room, so really appreciate all that you're doing to make this program possible. All right, the question was year in review. So this is a year plus in review. These numbers go through the end of last month. 216 families have registered so far. As of today, that number's a little north of 220. You can see. You can see that we do have a lot of international interest. This program is set up to help families being treated in the U.S. I'm sure you can also recognize that a family facing this diagnosis isn't going to stop a little line on a website that says this is for families being treated in the U.S., and so we do have quite a bit of international reach out. You can also see that over time, the average number of registrations per month is climbing, and that's good. And that's a lot because of all of you and what you're doing to help us get the word out. But we also know there's still a lot of families out there that don't know about this program or haven't gotten to the point where they'll actually make that connection. So we're going to keep working on that as well. I'm losing my place here. Talking about the program, we do a survey twice a year to be able to check in with families and get some feedback. And I'm proud to say that 96% have reported that they're likely to recommend the program. Quite honestly, I'm glad there's a missing 4% because it gives us the opportunity to hear about how we can be better. And so we listen to that feedback and we work on the program, and I'm going to tell you a little bit about some of those changes as we go on here. There's a quote from the most recent feedback about our wonderful Leslie Jared that I think really exemplifies the key message that we hear from this program. So I'm just going to read it.
Leslie has been a really great support. She is very passionate about this terrible disease, has a great deal of knowledge, and is also very compassionate and easy to talk to. She always makes herself available to either listen or help guide me through this ordeal. I'm grateful to have her as a contact and that people like her exist. And I can tell you we get similar feedback on all of our nurses, but that just gives you an essence of what we're hearing. Also quickly, we have received reach outs from 36 states and 33 countries so far. So, okay, I wanted to, just in reflecting back on the year, what are some of the key decisions that were made that has been beneficial for us from setting guardrails and enabling us to be successful? The first one was a decision to staff it by nurses. That wasn't an obvious at the beginning. We did consider a broader group, but in the end, as you all know, where you are, when you get that diagnosis, is that you need to be able to trust the resource fast. And being able to have experience in pediatric oncology was so critical, so that's what we stuck with. The next one was, we're not in the business of providing advice. We have plenty of really smart doctors who can do that. Our job is to help inform and support along the way, and that's been key. And part of that decision is we decided not to collect medical records. And thanks to our friends at XCures, we are either now or very, very soon in a position that our families are going to be able to choose to give our nurses access to their records. And thanks to that generous gift, we think we're going to be able to improve our experience with our families significantly. So very much appreciate that. The next one is the US. This is a hard one. Our nurses want to help everyone. The fact of the matter is, we don't understand the medical systems. We don't understand what is truly available for families that are not in this country. So we have made the difficult decision to draw that line. When a family reaches out, we do have an email that we're able to send them with as much information as possible and connect them with the resources that are closest to them. But that was a hard decision, but it is one we're sticking to for now. Medical interpreter service, we knew from the beginning we needed to be able to meet the needs of Spanish-speaking families. We have finally gotten to the point that we are fully able to do that. You can flip the website into Spanish thanks to Google and the little switch on it. When you register, you choose if you want to interact with us in English or Spanish. If you choose Spanish, at that point, all of the communication that goes to the family has been translated through a medical interpreter service. And then we're able to set up a Zoom where we have the medical interpreter service on that Zoom, and we can fully have the full experience through Spanish. The medical interpreter service is over 200 languages, by the way, but we haven't fully interpreted everything other than in Spanish. So we're on our way there. And then the last one is the important decision to separate this program from the general foundation activities. It is a separate website. The data is held in a completely separate system from our donor database system. It is incredibly important to this foundation that no family that participates in the program ever feels like there are strings attached or any obligation in any way. And you know, I know that's been something on people's minds, and we work very hard. In fact, I couldn't even tell you the majority of the families that are in the system. It's not something that I generally have access to. So that's these ladies over here. Okay, focus for upcoming year, awareness, awareness, awareness. Our ideal is within hours of a diagnosis, a family knows that we exist. And we're working hard on that, and thank you in advance for anything you can do to help us with that. Second one is engagement with the tumor board. You're going to hear a little bit more about that in a second. Our nurses attend every tumor board. They find it incredibly valuable. We feel very lucky that we're invited to attend that. And we have heard from the doctors that they find it valuable to have our nurses there as well. So we're going to continue to keep that as a focus. We are investigating expansion into Canada. We have a belief that we can meet the needs of families being treated in Canada. We are walking that a little bit slowly, but the hope is by the end of 2024 that we can figure that one out. We do have a leadership transition, which I'll talk about in a second. And lastly, if you look at the trajectory of the average registrations per month, there's probably a good chance we're going to need to expand our team next year. Back to those navigator partners, thank you very much. Okay, meeting our team. You will see these ladies up here in blue. We are missing Emily. You'll see her tomorrow. But we have Leslie, Ashley, and Lauren here today. I can tell you we have the best team possible, and it all starts with Leslie. Lisa was not wrong. And I think Leslie would join me in the element of it's really hard to say no to Lisa. So, but when Leslie joined us in 2021, she was clear that she was headed toward retirement and this would be her last song. And she would come and she would help us get this program up and running. And let me tell you, she got this program up and running. She is the reason it is what it is. She's the reason that these ladies were hired. She's the reason that they're as good as they are. And this just wouldn't be what it is without her. So we're very, very grateful. With that, Leslie has a wonderful woman sitting to her right in black here. Raise your hand, Daniela. This is our new director of nurse navigation, Daniela Ciccolini. I'm really thrilled to introduce her. Daniela is coming to us from Boston Children's Hospital, where she's a neuro-oncology nurse practitioner. She's in the process of getting her doctorate in nursing from Northeastern University. She already knows this world. She works with some of our favorite doctors. The ramp up is going to be very, very fast. She's going to be able to jump right in and help. We feel very fortunate that she's coming on board. So if you get a chance, we're around, our table's around, come over, say hi, meet her. We want her to meet as many of you as possible. So as a final wrap up, I just want to say thank you. Thank you to Jace. We're going to start there. He had the vision, and he knew it was necessary. And then between him and his mama, they weren't going to say no. This had to happen. It was not the most ideal time for our foundation to take on something like that. But it is probably the thing that I am most proud of in my entire career across all of my lives, professional lives, of what we've done and the difference that it makes. And it's because of you guys, our family partners, our founders, all of the people that were involved in the room, the researchers, our navigator partners. We together created this, and Leslie, I promise you, we're going to keep this thing going to live up to your vision. Thank you.
Hi, everyone. My name is Carl. I work at the University of Michigan, and I am here to give an update on our DIPG DMG National Tumor Board, which you just heard a little bit about, and I think it's actually really nice to come right after the Nurse Navigator program because these two programs have really melded together in a way that we hadn't even envisioned, but it's been sort of this synergistic benefit to put the Nurse Navigator program with the Tumor Board. I'll tell you a little bit more about the Tumor Board, but the nurses have come, and that's been really helpful for us, and I think for the families. They're sort of like a third party there that they can advocate if maybe the question that the family really wanted to know wasn't asked, they can just quickly chime in, and that's much more efficient than sending back a report and coming back. And I think the other thing is that in a short time, these guys know the eligibility for every trial better than the team of doctors. And there's a lot of times where we're like, yeah, we should, recommend that trial, and then, one of them will pop in and say, you're not eligible. Or did you think about this trial? And we're like, oh, that's, and so there's a level of knowledge there just from talking with all these families about the trials that they've become a really useful member of the Tumor Board. So this is not the full Tumor Board. This is some of the Tumor Board. And I like that you'll see a couple people with strong glasses on the bottom. Jason's coming in strong from Atlanta, along with myself. So, I know the theme has been you can't say no to Lisa. That is definitely what happened here. I think when she told us the idea, and I was like, well, I've never been on a meeting with some of these people at the same time because some of them are booking out 2.5 months. And she's like, no, every other week, maybe every week, I need you for an hour, hour and a half. And then I was like, I don't know if that's going to happen. And then she got Sabine, because we also don't say no to Sabine. Then the two of them, it was like this powerhouse. And the two of them wrangled everyone. They figured out a time. We moved our schedules. And that's what's happened for over a year now. And it's been really cool. I think we're learning what, in fact, we can offer to the community. One of the things that it is, and I think it's probably its strongest point, is it is a quick sort of shotgun approach of what are the trials that are eligible for this patient right now. And as you guys know, that is the most difficult thing, and that's the thing that takes sometimes traveling across the country, sometimes virtual visits, sending records. The records take forever to send. This is just one record being transferred to San Francisco, and there's a coordinator, Eileen, who's amazing. She gets the records, and then we run through the patient's story. Usually the physician who is directly taking care of that patient is on the call, presenting where the patient's at, and then what are the main questions. And then we go through MRIs on each of these cases, and I'll talk about that in a second. But then we go through and we come out of it with literally shooting across the, COG, PBTC, PNOC, institutional studies, focused ultrasound, CAR-T. There's 3 CAR-T centers. They all have different little nuances that are hard to tease out as to whether they're eligible or not. But you come out of that with, usually a pretty good understanding of whether you're going to be eligible for all those studies. And so that's what the clinician hears and then can go back to that patient. It's way more efficient than anything we were doing in the past. And I would argue there's not really anything else going on like that that I'm aware of across pediatric cancer or brain tumors, right? I mean, I think we're leading the field, and hopefully other tumor diagnoses can learn from us and create a similar platform. The radiology review has also been a really critical part of this that I guess I wouldn't have guessed would be so important, but just taking a second set of eyes, and it's usually a radiologist, there's 1 or 2 from Colorado and San Francisco that looks at a lot of DMG imaging. And so for them to see that imaging and just be an extra set of eyes, we often are seeing things a little differently than how they were interpreted. And a couple of times this has, I know a lot of families who say, well, we were going this route and then the tumor board looked at the imaging and then our team re-looked at the imaging and that's really changed our path. And so the sooner that happens, the better. So that's been another important piece. And it's also a lot of, it's 10, 15 physicians who are literally scrolling through the images, and sometimes they'll see something like, what is that spot over there? And then we'll look at that for a little while. So that's good too. I think those are the things that it is and what it's helped for. I think one of the things that it is not, and I'm not sure how to solve this issue, is that we often don't come out of that with these are the 4 trials and you should go on this trial. And I think this is a complex part that is maybe a little disappointing about the report so far, is like, okay, you just put it back on my shoulders. How do I know which trial? And we're trying to engage the physician who referred to be that driver with the family. But it's, we're running these trials because we don't know which one is the best. As you all know, we just don't have that standard of care yet. We have really good, promising trials, but we don't know which is exactly the right trial. And so I guess I want to get that word out there that as of now, what's not going to come out of that tumor report is you need to get on this trial right away. These are the trials that you're eligible for, and you need to talk with those teams and your physician about it. This is our enrollment from 2022 to 2023. And what you see across this is that it's a mix. A lot of the referrals are actually coming through the Nurse Navigator program now, but otherwise families are referring themselves. And then in terms of newly diagnosed versus progressive, it's a mix. We're seeing about half and half. This is going to go deep into the weeds on sequencing. So we are offering sequencing through the MoncaSeq program at U of M. The vast majority of these patients who make it to the tumor board have had a biopsy and usually have had sequencing. So that's about like 9 out of 10. But for those where we're seeing a case and it might be the first time that family's hearing a lot about, we really would recommend that biopsy. And if your center hadn't really brought it up, maybe you need to go to another center or at least meet with a neurosurgeon to consider it. But the other piece is, looking at the results, if we don't see that sequencing is done, we're for sure going to recommend it. And the DDRFA has funding for MyOncoSeq to do sequencing. So that is coming out for those patients that don't have that. I think it's going to go a little deeper into the sequencing than probably we need for this audience. Yeah. A lot of details. I'm not going to go through them all. Just as we were going through this talk, I was like, I'm not going to go through all those. I think what I really want to get across is that the program is thriving. It's very much supported by the DoD RFA is supporting it financially from a lot of directions, and this integration with the Nurse Navigator program is amazing. The referral system is working because as of a week or two ago, we just expanded the meeting from 60 minutes to 90 minutes. And so I think we're going to have this same growing pain that the Nurse Navigator program has, which is right now we're really following patients from the U.S. and what do we do for these non-U.S. patients, especially the ones in North America that might even be able to access these trials. So we're working on that growing pain, but the system is working, and we appreciate all of you for your support. I assume there may be some time for questions, but maybe not right now. Okay. Thank you.
All right. We're going to open it up for a few questions, and then the last speakers are going to be speaking on gift from a child. And so I just want to make that announcement because that is one of the most precious gifts our kids are able to give, the gift of tissue. But I always said when we were in the fight, some things weren't my chapter yet. And it's okay if after these questions you feel like this isn't my chapter or this isn't something I feel comfortable talking about. But we're going to talk about it because too often we get to those end-of-life days and then all of a sudden we're like, oh yeah, I definitely want my child's life to mean something with research. And this is a gift that only my child can give. And I know Jace, was older, but he definitely felt that way, which maybe gives comfort to some of you who have younger kids making the decision. But first of all, let's answer any questions you guys have with the tumor board or with my DAPG Navigator. And I do want to shout out to our DARFA members, the DDRFA. We came together to do the tumor board, about 60 foundations, and we stayed together, and we're funding research twice a year right now with research grants. And some foundations are brand new, and they might have $10,000 to give, maybe this year. But together we can make really big impacts, and then others are bigger. And we're part of Tough Together, for instance, is part of Chad Tuff's Navigation Partners. Chad Tuff is part of DARFA, so we're all working together really well. I also just want to say thank you to Sarva, who is our membership coordinator for DARFA. So if you're interested, you want to get with her this weekend. And Misha Mehta, who is our industry liaison, who not only do we fund grants, but Misha goes into industry and says, they need these assays. Can you do those for free for us? Or can you do these at a discount? They need these lab supplies. Can you do this? Which makes our money go farther. So it's, again, ingenuity of people in this crowd that make it happen. What questions do you guys have about Nurse Navigator, the tumor board? Brandy Lee, I see you have a question. They're bringing you the mic. Yeah.
I don't know if I missed this or not, but I wanted to know if the nurse navigators, if this is your primary job or if you have another job. You are, okay. And so they're fully supported, funded, have jobs, and this is their only job.
Yes, so we have currently 3 full-time. Leslie's part-time as she's heading herself off to retirement. Daniela will be full-time. So they work remotely from home. Lauren's in Indiana. Daniela will be in the Boston area, or is in the Boston area. Ashley is in Alabama, and Emily, who will be here tomorrow, is in Tennessee.
Just raise your hand and I'll come find you with the microphone if you have a question.
Hi, my name is Mairead Finn. I have a question with regards
my navigator or my DIPG navigator and collaborating with XCures, is it going to be a case that the nurses would actually be able to enter data in of the patients, or will it be the patients themselves that will actually enter that data in?
It's the patients themselves. So basically, our navigators are being set up as a provider group, and we'll be sending our families with full explanation as to what's going on and what they're consenting to over to XCures, and then everything happens through XCures. Then the family has to say, yes, please give the nurses access to my records.
And we might let XCure speak to that, but they do the automatic job, which brings them in a matter of hours. What? You just have to sign up.
We do all the gathering of the medical records. You just have to go online, fill out the form, and the
And there is no cost to you, and it stays in a secure portal. So if any of you in the crowd had trouble with my chart turning off at some point, you're not having access, this continues, and you can share it with anybody you choose to forevermore. So while you're in the fight, if you're taking an amazing trip and something happens, you've got access to everything. If you're not in the fight, but suddenly you're like, I just want to relive that and make sure I remember it correctly, you've got access to it. And DARPA worked with XCures to make sure we had a data navigator, so someone who can visit with you about your questions at any time. And they have all kinds of experts at their disposal. They have an amazing advisory board with genetics experts and imaging and everything. So really good crew over there. But won't take the place of our tumor board. Let me say that with our DIP-DMG specialist. These are the guys and gals that you want. Sandeep, up front. I don't know if I should repeat that in the mic. He asked if I could convince his wife to slow down because she's turning into me. We need a whole room full. We need a whole room full of Mishas is what we need. Sandeep, how do I convert you? There are a few gaps that we see as patients, and sometimes we see them differently than clinicians. And clinicians, Carl, you probably see gaps we don't see. But one gap we want to make sure we understand is, and in early, like as soon as you're diagnosed, be able to find it. What should your biopsy include? Because not all biopsies are created equal. Do you want to speak to that for just a sec?
Yep. So one of the things that I tell families that I often see in consult is they have a diagnosis which comes just from staining the tumor tissue and looking under a microscope. And that can get you a DMG diagnosis, especially if they see the histone mutation. And that, quite honestly, is how far it goes for a lot of these cases. But really what we'd recommend is that you go that next layer lower where you're looking at the DNA of the tumor and you're comparing it ideally to healthy DNA in that same patient. That's the germline. So that's the buccal swab or a blood sample. Beyond that, most of the platforms, as long as they capture the pediatric midline glioma mutations, which they're increasingly doing, are going to get us 95% of the way. I think as a parent, or especially if you're just going through this, you would have no idea to ask for that. And so one of the benefits, I think, of coming through the Nurse Navigator program or through the tumor board is that they're going to look at those and just give you feedback on, did we get a full view? Do we have enough? Sometimes it is in those sequencing results that we can be able to give a little more guidance on, like, this is not a great fit. We know that type of genetics is not a good fit for trial A or B. And we increasingly know some of the genetics, in addition to the histone mutation, are going to drive prognosis. We're getting better at being able to predict a little better when to expect progression. And that can be hard information to get. And so I think it takes the right clinician to sort of sift through that. But I think we're moving in a direction of needing more and more information, not going with less. And fortunately, I think the tools are getting better.
Awesome. And I think we're probably going to see maybe like this is a standard best practice, what you should ask for. I always cringe when somebody calls me and says, all they told me was H3K27M. It's the worst. And you're like, okay, but there's more, right? So we want to get that more standardized. With the DIPG One Link, we give parent advice, which is not medical advice, but sometimes you forget to, our clinicians forget to tell us, you don't have to go to a special center for radiation at this point. Is that true? That's a place you can spend those 6 weeks close to home. But it doesn't mean you necessarily want you to do your trial close to home if there's a better fit for a trial. So we want parents to understand, like, you can't put the covers over your head. You do have to hit the ground running, but there's a navigator, there's a tumor board, there's XCures to start to gather your information. You do not have to do this all by yourself. We also want people to ask when they do a biopsy, can I have the results to share? If we're putting all the data into something like XCures, we can't have those biopsy results be research level results. We need them to be able to go into industry and be able to be aggregated with the other data. So more and more that's going to be an issue, and that's an issue that our clinicians are working with us on because it challenges some of your institutional norms too, right? So we have to figure that piece out. But it's important because if I can't take my results outside the clinic and be able to do what is commonly done like at Cancer Commons, which is they send you all kinds of published information and options. Sometimes when you're out of options, you want to know what those are. It's only going to be helpful if they can analyze those results. Okay, one more question, and then I want to turn it over to Jenny and Melissa. I've got one in the back. Two more questions. I can't say no to Milan.
My question's about the Navigator. How are you guys advertising it more than just brochures? How do people know about it?
Yeah, great question. So we have a few different avenues. So we have an effort going on from a Google ad perspective to try to be there from a search standpoint. So, you get diagnosed, you're going to go to Google, what's DIPG? We're trying to get Navigator to be the answer.
Great.
That's a key. We also did a direct reach out, and we plan to do this annually, where we were working together with the tumor board to try to get this information into the hands of pediatric neuro-oncologists across the country. Those are really the two main ones. The other piece would be we are active in the Facebook group, the DIPG research, I never remember the name of it. Right. And the administrators there have been so supportive that when we see families that are there that are just diagnosed and they're from the US, we're private messaging them. We're trying not to look advertisement-y-ish. So right. My nurses will let me know. I'll private message them of I just want you to know about this system, and we're here to help you. So those are the primary ways, but if people have other ideas, come talk to me.
As a nonprofit founder, I have a link on our page, but what else can I do?
The best thing is when you find families, and especially on social media, just send them to mydipgnavigator.org. Thank you.
Yeah, and I think the DIPG One Link too, because sometimes people are just not ready to talk to somebody. So I think between the websites, we cover a lot. The other thing to know is we have a private community forming, the Tough Together online community. It's on the Hands-In platform. It does not collect your data. And there, many of us will be standing up small communities. So what do you do when you get that tumor report, and there's 4 options that might fit? Well, maybe you go into the community and find out, like, how are kids doing on these trials? Like, what do parents say the advantages or disadvantages are? It's helpful, but we have to remember that what happens to my son might not happen to your daughter or the next, right? These are small, early trials. They're just seeing if it could possibly get to the tumor, and then if it's safe enough, and then we find out if it's effective. So we have to kind of know those phases as we get into clinical trials, and we talk about that on DIPG One Link.
Yes, I think I was going to ask the exact same question as to how it's being promoted. So you did talk about it, but besides that, what happened is, I ended up talking to my kid's pediatrician and told him about this, and they were willing to put flyers of my DIPG in their office and in their facility. So that is what I gave it to them, and that would be something that all the parents who have the kids or had the journey of DIPG earlier, it would be something that they can do to talk to their pediatricians and say, can you just put up a flyer? So that would be a second way of reaching out to doctors nationwide.
Yeah, that's great. And we have a digital version. So some of our families are sending the digital version of the flyer to their doctors. But I'll work with Lisa, and we can put a way for people to send us names, and we'll send, and especially because we love doing it with the tumor board together with Navigator, and we can send those to anybody that, here, send them to these people. We'll stick them in the mail.
And guys, what if we did this? What if we honored our kids by the date of their birth date every month? We just post on our own Facebook pages. We're so proud of what's moving forward. We want you to know about my DIPG Navigator, about OneLink, about the tumor board, because I know it's happened to you and it's happened to me. All of a sudden, somebody that you know, a friend of a friend or a cousin of a brother, has DIPG. So awareness matters, and you can always say, in honor of Jace's birthday on the 2nd, would you join me in posting so that families that face what we faced don't do it alone? I think that would be a way to do it. I want to switch gears just a minute, because one gap we have not solved in communication, we haven't solved it on the clinician side, we haven't solved it on the parent side, is when and how we have a discussion about tissue donation. Now, many of us on our driver's license say, oh yeah, I'm an organ donor, right? And it doesn't mean we think we're going to have to donate our organ tomorrow. It just means if ever we found ourselves in that situation, we want to be able to help someone else, right? So what's important, I think, is that we start to change the conversation from something we're afraid to talk about till the end of life, and we start to have it along the way. Like, these trials are possible because we learn from patients, and that's really the way we have to learn. This data collection is important because it's the patients who can tell us the most. I can't imagine what it's like to be a clinician and have to say, there's just nothing more we can do for your child, but if we can have their brain, that would be great. Like, that's not the right conversation. So we, as parents, need to help guide that conversation. And that way, we'll get to the point we have many, many survivors. But we have a really important tool, and it's through Gift from a Child. I've worked with them at least 30 times, maybe, in the last year and a half, very close with their navigators, and many of you have worked with me in it too and have made that hard decision. And it is heartbreaking to have that conversation in the last day. So I wanted Jenny and Melissa to just share a little bit with you. And again, if you're like, I didn't get the chance, this is too hard for me, or I'm not in this chapter, you will not offend us. But just know that if anyone you know ever needs that information, I think any of us at the table, maybe the nurse navigators, maybe Ann, are happy to have the discussion, because we also know parents that say it's in some way given a purpose somehow to the pain we've gone through. I feel that way. And I also know parents that are furious that the one thing they could have made a decision on that could make a difference. We don't get to make decisions a lot of times if our kids can enter a trial or not. But the one thing I could have done for somebody else, nobody told me about. So I want to take a few minutes to explore that with you. Yes, you do. Yes, you do. My slides coming up. Nope.
I think you had to wait for them to flip it.
Okay. Thanks, Lisa. And I couldn't have said that better. And we had a session a few minutes ago, and we heard these stories, these heart-wrenching stories of families, but intertwined in each and every one of them telling about the loss of their child was a little bit of hope and a little bit of, as Dr. Mark taught us, bereavement care. And so thank you for staying and listening to this, because you asked how you talk about the DIPG navigators and the tumor board. And it's each and every one of us telling our story and telling what it meant to us to use any of these services. And it also means talking about what donation may have meant to you. I'm going to quickly talk about Gift from a Child, and then Dr. Mark is going to talk about what it's meant at her hospital, in her institution, and in her practice. Gift from a Child has 7 centers of excellence across the country at some of the premier hospitals. We have at University of Michigan, in the Koshman Labs, a Gift from a Child center as well. We were very excited to start working with Dr. Koshman. And we also bring in referral hospitals. And our referral collaborators are hospitals that don't have the facilities to do their own procurement and donations. And our navigators at each of these 7 centers work tirelessly to navigate for a family, on a family's behalf, and on their care team's behalf, to make the wish of tissue donation a possibility. Because we want nothing more than the families to spend every minute they can with their child as they're nearing end of life, and we don't want the burden to be on the physicians. Our navigators, like the My DIPG navigators, our tissue navigators, put the family first and try and make every wish possible for the family. Since 2019, and as of August 31st, we had facilitated 280 donations, or 260 donations across the country. 142 of those donations were outside of these 7 centers of excellence. That represents over 75 institutions across the country who may not have been able to help a family make this precious gift, facilitate a donation because our navigator stepped in to help them. Our resources quickly, we have tissue navigators who will work across institutional and state lines. We have a family companion program. We will connect you with a family who's been in your shoes so that you can ask the questions that you might only ask another parent. Gift from a Child has resources and stories to help you make a decision. Print materials. We have a video online toolkit that will help physicians have this conversation, this very difficult conversation. Very little information needed. We're going to be sharing these slides, so I'm not going to walk through these because I really want to get to Dr. Mark. But there's 3 consents. Every donation we take, a portion of the donation is donated to CBTN so that it can get into the open portal sharing. So that's my part of Gift from a Child. Dr. Mark is going to tell you how she has used our resources to build a program in her center.
You just have to wait for them to flip onto yours. Give them a second.
Hi, I'm Melissa Mark. Nice to see you. I'm really honored to be here and speak in front of all of you. So, I'm a pediatric neuro-oncologist at Children's Hospital of the King's Daughters, which is in Norfolk, Virginia. It's about 3 hours south of here. I am just at a regular children's hospital, so we're a medium-sized institution, one of only 2 freestanding children's hospitals in Virginia. We have a robust neuro-oncology program, follow about 200 patients. We don't do, I'm not a researcher, I'm primarily a clinician, and it's really important to me to bring clinical trials and opportunities like this to all of our patients, and I've had the pleasure of working with the DIPG navigators and the tumor board and Dr. Koshman. And so all of you as parents know how important tumor tumor tissue is to research. And really, it's parents who make this happen. And we know that not only is this important for research, but it's also important for our legacy and bereavement for our patients and families. And really, Gift from a Child has done research to show this. The barrier is not parents, the barrier is clinicians. And so we really want to spread the word to empower patients and also empower clinicians that you need to have these conversations, you need to find the words. And we have all kinds of difficult conversations as clinicians, and it's really important that this is included as part of this continuum of care for brain tumors. So if you come to our session tomorrow about palliative care, you'll see that opportunities for tumor discussion really follow opportunities to integrate palliative care. Tumor donation is not something that needs to be talked about at end of life or after death, and in fact, those are the most difficult times to talk about it. So we start planting seeds and talking about this from diagnosis. So just like Lisa said, when we talk about clinical trials, we say this trial is possible because families have donated tissue, and that's that, but then that's there and families know about it. Other opportunities where it comes up where families are naturally curious about it is at progression or a change in clinical status. Advanced care planning is also a big time where this comes up. So if you're doing some kind of advanced care document with an adolescent, for example, Voicing My Choices is a common document that many people use. There's actually a page in there where the adolescent can elect and say, I would like to donate my tumor. Well, as a clinician, if you're not at a research center, you better know how you can honor that wish. And Gift from a Child is here to support you and support those logistics. End of life is often a natural time where it's brought up, and it can be discussed after death too. A lot of clinicians, if a family wasn't interested in a trial, didn't have an advanced directive, didn't have a DNR, are really hesitant to approach those families. And in our experience, even in that situation, families have been willing to donate, and it has been an important part of their journey. So this is just a look at my data from our center from last year. And I just show this to say the spectrum, there's no typical tumor donation patient or family. The spectrum of disease is varied. This is not just for DIPG, this is not just for DMG. And all of these conversations were had at different points in the patient's care journey. Our goal is to provide goal concordant care, and tumor donation goes along with that. And when you have that discussion is based on the goals of the family. Also, the location of death does not impact a family's desire or ability to donate. So lessons learned from our center, and again, this is really for any clinicians in the room and any families to bring back to your clinician, is that I really challenge every clinician to offer this as a standard of care for all patients and families. If you're going to pick and choose who you think might want to donate, you're missing out, and it's not fair to families. We know that families who don't have the opportunity to donate express regret, and we know families who do donate have better bereavement outcomes. So this goes beyond just the need for research. It impacts bereavement. And for our patients who choose to donate, we know that it impacts their sense to build a legacy, their agency, and their meaning making of their brain tumor experience. Things that we have done to support our families, the logistics have worked best for us to use the funeral site as the site of procurement, just so there's less moving around. And we have decided as a team to have a team member present at the time of procurement for all of our patients. So I'm either there to oversee the procurement and assist with the third party who's doing that and update families in real time. Hey, I'm here. Hey, we're starting. The courier has it. Your child's tumor has arrived in DC. The research team is, extracting tissue, starting cell culture, et cetera. Another thing that we have done and are able to offer to some families is to meet with the research team where the tissue has gone about a year later. The team is able to give a real-time update, and then as the clinician, I sort of facilitate as a translator sometimes in helping that PhD and research team having these conversations with families, teaching them how to talk to bereaved families, encouraging researchers to use the child's name, and we've had a lot of really great outcomes with that. And it's also given families a chance to come back, see their team again, and have ongoing bereavement support that way. So we're happy to take any questions. You can reach out to me. I'm happy to talk to providers and give them more support, to give them the words to have these conversations with families, and can't speak enough to all of the support that Gift from a Child has given me at my institution in making this happen for patients and families.
Does anyone have any questions for Ginny?
Yes, it's not more of a question. I just wanted to share a short story because my son, Ronnie, he passed away in 2018 when he was 14, and was diagnosed when he was 12. So, I mean, it's really hard to even talk about it right now, but when he was 13, he was the one who started talking about that, you know what, I need to do tissue donation. I had not even thought of it as a family. And he made plans for it. He reached out to Stanford while he was, going through his journey. And Dr. Manji said, okay, we'll set it up. And then when he passed away in September of 2018, so he did the donation. And 2 years later, Dr. Manji, she said that what he had was the Wild Type Foundation for H27K. And his was the only tumor in the world that was viable to be as a cell line to be used worldwide. So, and there's a lot of trials going on for not trials, but research going on for wild type tumor using his cell line. So I just wanted to say that, his legacy lives on, but, if as a family you're thinking that, I don't know what's going to come out of it, even we didn't know. But my son, he thought that, let's just do it because we never know what can come out of it. So that's something that, we can just try to think and say, yes, it's for the future kids. It might help. Yes, yes, and I think his tumor now has a name, and that's what gives us, kind of feel proud about it, that even if he's gone, it's like he's called SUDIPG48 in the DIPG world.
That you can ask. At Stanford, they will name theirs one way, Michigan maybe another way, but most institutions will share your identifier for your sons, and I know many of us have seen those in publications, and if that's what you have left of your child, there's a lot of pride in that. And I would be remiss not to tell you that Gift from a Child came from a young teenager who felt just like this. And Michael's legacy through the Swifty Foundation and Gift from a Child lives on far, far longer than his young life. But he absolutely knew, as I understand from his parents who are in the back of the room, that this would be his legacy, that this would be important, and he would help other children, and he has. And he has, and that's something we just need to keep getting the message out. None of us want to be in this situation, but none of us want to not have the choice and 2 years later say, what if, just what if my child's cell line had grown in a way that would inform research. So this is delicate, but we have been through this. And if we have been through this and feel it's important that our clinicians know that too. And today we've talked about DIPG as a continuum, a patient continuum, because that's the way we see it. And we see the DIPG One Link as putting that continuum all together. And it will continue to grow and be more beneficial through the discussions in the online community. So I encourage you to go. If you've lost your child to DIPG, DMG within the last 5 years, go on the DIPG One Link, find X form, and go ahead and consent and pull that data together. And then you can continue to track what comes of it as well. And I will say, since the announcement of the White House last week about DARPA creating this and highlighting this, I mean, I've had 3 calls from companies who said, we're standing up a glioblastoma trial, or we're standing up a tumor type that has the H3K27M. And if we could just have enough data, I think we could do an arm in DIPG. We'd be interested in that. But we need the data to know, do you think we can get it? Well, that's exactly what we're trying to do. So you will all be a piece of that story and sharing that story, and we'll only make it better with all of you in the room helping to fund, to provide data, and encourage others. And I hope that the best practices of it could be a model for other pediatric brain tumor types, because I think it has given all of us just a point of pride that the next family will face it with just a slight bit more ease. All right, thank you so much. We're going to go into a fun time, a reception. We get food, and they're going to open up these doors, and you're going to have some cocktails and food as you choose. They're going to leave this one open so you can see sponsors. Oh, is the food in there?
Come visit our sponsors tables first. So as we're leaving here, please come check out all these amazing tables out here. And in that time, we'll open everything up and get it ready for us.
And
So
just,
visit our sponsors.
Just housekeeping, if you have children upstairs, they can come down, have food as well. You're going to want to feed them. At 7 o'clock at 5 Iron, we start Guys Night, and that's going to be a blast. At 7 o'clock, right in this room, we're going to stay, and we have Gals Night in. So, and then the kids will have movies and video games and fun stuff back upstairs. Thank you all for the first day. It's been wonderful.